All righty. Today is Wednesday, September 8th. It always gets scary when the schedule gets short, but that is indeed where we are at. We have our last major concept that we need to discuss today, and that is going to be the physiological process of protein synthesis. And that's all we're going to talk about today. It's our last major concept, and it shouldn't take the whole class time, which should leave us with the opportunity to do a review for our first exam. As I mentioned before, a review is not me standing here saying the things that I think are important. That is what I do every day in lecture. Instead, a review is an opportunity for you to ask questions about the concepts that we've talked about in this class, things that you're unclear about, things that you don't understand, things that we can help you to be successful with when we get to the exam. And remember, if you don't ask questions, then I assume everybody has mastered the material and I make the exams harder. And that is what we're doing on Monday. Remember, if you look at the schedule, you will see that technically we have a class slot time for lecture, but there is no lecture. You are going to take your lab and lecture exams. Again, remember the rules for this. They are timed exams, meaning that you have a set amount of time to be able to complete them with. I have not finished writing the exams yet, so I'm not sure exactly what that is, but usually it's somewhere between an hour and a half for the lab exam, and somewhere between 2 and 2.5 hours for the lecture exam. However, remember, they must be completed. During the class time. So remember what that means. If you have 2 hours, timed for the lecture exam, but you start the lecture exam at noon, you're only going to have 35 minutes to complete it. So both exams must be completed, not started, but must be completed between 8, that's not an 8, there you go, and 12:35. So 12:35, the exams get shut down and you will no longer have access to them. So again, make sure you complete them during the time. You should have plenty of time to do that. Again, you can complete them in any order you want, but I encourage you to A, start early, and B, to start with the lab exam. I would say that 90% of the problems that students have had with the exams have involved the lab exams. Again, I've tried to minimize that by having the pictures load one at a time, but still some people do have problems with it. So it takes some time to work those things out. And again, you have to fit this all within our class time. So you're not required to start at 8, and you're not required to start with the lab exam. You can do it in any order that you want. But most people that have had problems, they have those issues like that. So again, if you wait till 11:30 to start the lab exam and you only leave yourself an hour to complete it and you have problems, then you're going to run out of time. So again, I encourage you to do that. If you haven't had any problems with Proctorio, good. Hopefully that won't be an issue, but especially after the practice lab exam, now that you've seen what that looks like, if you did have problems with that, hopefully we've resolved those. And we can go from there. All right, that is the game plan for that. But so like I said, there is a lecture time, but we're not meeting for lecture. Assuming everything goes well for you on the exam, you won't see me and I won't see you. So that is just for the exams. Normally, I don't like to look too far ahead, but the last thing that I want to point out to you is because of the way the holidays fell out this semester, our second section of the class, where we talk about tissues and we talk about the integumentary system, usually are about 3.5 days worth of lectures. Some semesters I have 4 days to work that, and so we're able to spread it out. And some semesters we only have 3, and so we have to pack that information in. This happens to be one of those semesters where, because of the holidays, we only have 3 days to cover this material. So again, it is going to be a very fast turnaround, basically 2 weeks to cover this material. And for many people, it is some of the more challenging material, because while many of you have probably heard of the brachial region before, the femoral region before, or know what anterior and posterior mean, not as many people are as comfortable with the tissues. So it doesn't hurt after Monday, when the materials are released for the second section of the class, to start looking ahead and thinking about the material that we're going to be covering, because we've got a lot of material to pack in a very short period of time. All right. Questions on any of that? Excellent. I so enjoy my stunned silences first thing in the morning. Excellent. All right. If there are no questions on that, then like I said, let's dive into the last concept we have to talk about. And that is going to be our process of protein synthesis. As we talked about, we know our DNA codes for genes, has the information that allows us to produce a protein. And when you take biology classes, one of the rules that they talk about is one gene produces one protein. However, we can be a little bit more sophisticated with that, because as we talked about when we talked about that structural organization of proteins, we know that most proteins are made up of multiple subunits of amino acids put together. And remember, the name we had for that subunit was a polypeptide. So for instance, hemoglobin is made up of 4 polypeptides that are bound together to make a functional protein. So in reality, a gene really codes for the information to make one of those polypeptides. And then, as we know, those polypeptides get sent to the Golgi apparatus to be cut and folded and modified and turned into a functional protein. Now, this process of making that protein basically, or really making that polypeptide, requires 2 steps. The first is transcription, right? Again, we have this issue. The information to make the protein, that DNA, where is that located again? Where is DNA located?
Nucleus.
In the nucleus. Excellent. It's located in the nucleus. Where do proteins get made? What was the structure, the globular structure?
Ribosomes.
Ribosomes, absolutely. And where are most of the ribosomes located?
In
the rough endoplasmic
the rough
reticulum.
endoplasmic reticulum.
Right. I'm going to abbreviate it. Know that you should not do that on the exam, but absolutely in the rough endoplasmic reticulum, which, as we mentioned, is in the cytosol. Right. And so we have a problem. We have this DNA inside the nucleus, and yet the things that make the proteins are outside the nucleus. So does somebody show up with a library card, check out the piece of DNA that we need, they let it leave the nucleus, go out to the ribosomes, and then they return it in 2 weeks, and if they forget, they get charged a nickel fine a day and that kind of stuff?
No.
No, that DNA is locked up there tight. I always, in my mind, think of it in terms of like medieval times. Knowledge was power. And so things like books were kept in places like castles or places like churches. And again, could you show up with a library card at that church and just check out any old book that you wanted from that church?
No.
No, absolutely not, right? First, they had to deem you worthy. And then if they were deemed you worthy, they would just give you the piece of information you were asking for, not the whole book. They would make a copy of it. And of course, who were the people who were responsible for making those copies?
Transcribers.
They were scribes, absolutely. The scribes were the ones that were responsible. And one of the really interesting things is most scribes in those medieval times were actually illiterate, right? Because as I mentioned, knowledge was power. So basically the scribes couldn't read. Their job was to look at the information on the page and just make a copy of it without fully understanding what it was that they were looking at. And that's what we're doing here. This process of transcription is all about making a portable and temporary I didn't need to all be in caps, but it's all right. Making a portable and a temporary copy of the information. Something that was capable of, no, great question. Something that is capable of leaving the nucleus. It is similar to what is happening in S phase. Remember, S phase, we're also making a copy of the DNA, but there we're making it in DNA. It's staying in the nucleus so we can have a new nucleus. This new temporary and portable copy is actually RNA. So they're both nucleic acids, but they're different types of nucleic acids. So you've got the right idea. It is definitely similar to what happens in S phase. So we have some familiarity with it, but it's not identical. Yes, Augustina.
So is this happening during mitosis in all of those processes, or is this completely different?
So this is in many ways, this is the opposite of mitosis. Remember, in mitosis, all the energy, all the resources, all the effort is going into the division of the cell. The rest of the time, when the cell's doing all of its normal functions, what primarily lets the cell do its normal functions is the making of proteins.
Okay, so this is during G0.
G0 or interphase, absolutely. Again, we've actually talked about this, even though we haven't really thought of it in those terms, because if you remember, one of the things we talked about is when the cell is in interphase, when the cell is in that G0 phase, when the cell is doing its normal jobs, this process of making this temporary portable copy of the genetic material requires a tremendous amount of resources. So many so that they condense around they condense around the DNA, forming a structure that we can see in a light microscope. This process of transcription is occurring in the nucleoli. So those nucleoli, those dots we saw in the nucleus, is actually where this transcription is taking place. So there in the nucleus, really where that nucleoli is where this process is taking place. Now, let's say we could go back in time and you were able to go to that castle and they deemed you worthy and they made you a copy of that information and gave you the information. Would it really do you any good to get a copy of that book that's in that church? What do you think? If I skip back in time, got a copy of a page from that perch from the scribe, came and brought it to you, would you be able to read it?
No.
Maybe.
Why not? Maybe. Based on what?
Language.
A type of language.
There you go. And what we know of medieval times, most learned individuals who wrote most of the books, what language did they use to write those books? Latin. There you go. How many people here speak Latin? Anyone?
No.
No. So that paper wouldn't do you a lot of good. What would you need to do to that paper first before you could use that information?
Transcribe it.
Translate it, exactly. And so that's the second step in our protein process. Translation is where we change the language of the information. We have to go from the language of nucleic acids and what is the language of nucleic acids? What are nucleic acids made up of?
Nitrogen, carbon.
True, but what do we call, no. Proteins are made of amino acids. What are our nucleic acids? What are the building blocks of our nucleic acids?
Peptides?
No. Remember, it had a sugar, it had a phosphate, and it had a nitrogen base. What did we call that?
Oh, a phospholipid? No.
Nucleotide?
Ah.
It's always the simple answers.
Yeah, exactly. So we have to change it from the language of nucleic acids, the nucleotides, to the language of proteins. And you guys already mentioned what that is. That is amino acids. So we have to translate the information. And so that's our process. We have to go from the information of the DNA to the information of the proteins. Change it from nucleotides to amino acids. And then we have our polypeptide, which, like I said, at this point, we then ship it to the Golgi complex. And once we ship it to the Golgi complex, it's cut, folded, and formed into a functional protein. So our goal here of this protein synthesis is really to make that polypeptide, to make a sequence of amino acids that can then travel to the Golgi complex. So that's our goal, protein synthesis. And here we've got the pretty picture that kind of shows that process. We're going from the information of the DNA, making a temporary copy, sending it to the ribosome, making a sequence of amino acids. And that is our job. That is our goal. So we need to talk first about transcription. In transcription, remember, our goal is to go from the permanent DNA to the temporary RNA. But notice both DNA and RNA, while there are some differences, are essentially the same language. They're both nucleic acids. Again, both of them are made up of 5 carbon sugars. Of course, one of them is a deoxyribose sugar, and the other one is a ribose sugar. Both of them have phosphates, those really negative structures that we talked about. And both of them have nitrogen bases that can be bound to them. How many total nitrogen bases are there again?
5.
Excellent. And just for simplicity, give me the letters of them.
A, C, U, G, and T.
Excellent. Do both DNA and RNA use all 5 of these?
Yeah.
No.
No. How many does DNA use?
4.
And which 4?
A, C, G, T.
Excellent. And how many does RNA use?
Uses 4.
Excellent. What 4?
A, C, E, U, G.
Yeah, G and U, exactly. Basically, the big difference between one of the 2 big differences between DNA and RNA is which sugar they're using and which of these pyrimidines they use. RNA uses uracil, DNA uses thymine. So if you remember when we talked about a strand of DNA, if I gave you a strand of DNA, right, as we talked about, you can give me that complementary strand that would go along with it. So if here we had a strand of DNA, like the template strand, let's do this. What would the complementary strand to this be?
U.
I know with DNA, we're going with DNA. So it would be T.
D,
Oh,
G,
T and T.
A,
G, T, G,
Whoops.
C, A, T.
There you go. Excellent. And that's the complementary strand. And so remember, in our process of replication of the DNA, we basically split them apart, and using our complementary base pair rules, we were able to make new additional strands. And remember, as we talked about, there were dozens of enzymes involved in that process, but the primary one was DNA polymerase. Well, here we are going to do something similar. We need to make a copy of it, but we need to make a copy now in RNA. And so for that, we need, again, dozens of enzymes, but one of the primary ones is RNA polymerase. So DNA polymerase helps us make more DNA. RNA polymerase helps us make RNA. See how convenient that is? And this process is basically a three-step process. Now, as we talked about with DNA, we know there are the complementary base pairs. As we talked about, we know that adenine can only bind to thymine because it forms 2 hydrogen bonds. Guanine can only bind to cytosine because it can form 3 hydrogen bonds. But as we mentioned, for RNA, we switch it. Whereas DNA uses thymine, uracil is used for RNA. And you can see they're very, very similar pyrimidines. So that's why they're able to be interchanged. And of course, both form 2 hydrogen bonds. And so both of them are able to bind with purine adenine. Now, this process of transcription is a 3-step process. So let's talk about those 3 steps. Now we need, first, we'll start easy. We'll start with our strand of DNA because it's convenient. So this is a small portion of the gene we're interested in. So as I mentioned, this would be what we would call the template strand. Now, as we know, there is also a complementary strand. And you know what? I'm not even going to bother writing all of it. Let's just cheat and do this. Although we can do a teeny bit of it. How would it start?
G A C C C T T T.
Yeah, G A C C C T T T A A G C, and so on and so forth. Right? We understand and get that base pair rules. All right. So we are talking about transcription here. And as I mentioned, this is a 3-step process. The first step in this process is actually kind of a serious one. Someone remind me again how many strands of genetic material we have inside of each of our nuclei?
2.
2?
And our nuclei?
46.
46, absolutely. 46 strands of DNA that are that loose chromatin that are going to fold up into chromosomes when it's time to do that. Each of these individual strands of DNA are comprised of thousands of genes. All right, so we have thousands upon thousands of genes on our strands of DNA. However, even with all of those genes on those strands of DNA, it only makes up about 1.5% of the DNA inside our nucleus. There's a massive amount of what used to be known as junk DNA, a bunch of repetitive patterns that seem meaningless. There's a whole field of study looking at the epigenetics of what the implications of these things might be. We don't care about that right now, but what we have is an issue. If only 1.5% of our DNA codes for a gene, and I were to take a dart and randomly throw it at the DNA, what are the chances of me hitting a gene, let alone the exact gene that I need? My chance is pretty good.
No,
No,
slim to none.
yeah, exactly. So our first issue is we have to find the right place to begin. That step, that first process, is what is known as initiation. Initiation is basically where we begin the process. We have to find the correct starting point. And luckily, to do that, we have a special sequence of DNA. This special sequence of DNA is typically something along the lines of like AAATTT. This is that special starting sequence. And when our special protein, that enzyme, let's do it this way. Our special protein, that RNA polymerase, finds that starting point, it is able to bind to the DNA, and it temporarily separates the sequences, the strands of DNA. So our RNA polymerase binds to that region, and then it starts to separate the strands of DNA. However, this is a temporary separation. Unlike in DNA replication, where we pull them apart and they stay apart and we use them as templates for making new strands of DNA, at the end of this process, the DNA is completely unchanged by the process. So what happens is our RNA polymerase pulls the strands apart. And then the same way we did using our base pair rules, it is going to start to make a sequence of nucleic acids, but it's going to do that using RNA. So what would the RNA base here be? That would be the equivalent to C, the complementary to C.
G,
G, excellent. And then what would be next?
C,
Excellent. What would be next?
A,
And then what would be next?
U.
Excellent, right? So notice using our RNA complementary base pair rules, we would continue down this line. And would this continue forever until it gets all the way down to the end of our DNA strand and falls off the end?
No,
No. So while it gets that starting point, the initiation zone, that initiation sequence, it is then going to, this process of adding the bases, I guess I should have added that on here. This process of adding the bases is step 2 in the process, elongation. Notice also we are only, why am I not writing here? That was frustrating. All right, let's try this again. So in elongation, using RNA base pair rules, we make a copy of the template strand of the DNA. Right, notice we're not doing anything to the complementary strand, but using those base pair rules, we're going down the line, making this copy of the DNA. Yes, Augustina.
Can we go over like why it needs that special sequence of like where it's starting again? It just didn't really like make sense to me.
Because we need to know where to begin. As I mentioned, only 1.5% of the DNA that's in a nucleus codes for a gene. So we can't just have our RNA polymerase just randomly binding onto the DNA someplace, because if it just finds a random spot to bind onto the DNA, is the part of it that it reads likely to be meaningful?
No.
No. So we need a way to say, hey, right here is the gene you want. Come start right here. And that's basically what it is. It's telling the RNA polymerase, this is where you need to come and start the process. Bind here, because this is going to give you the information that you need.
But how does it know like where exactly the place is?
The short answer to your question is that there are other enzymes and chemical signals that are going to tell it where to begin precisely.
Okay, so our own like enzymes and those things, they know what's the junk DNA and then the actual meaningful DNA basically.
Well, that's what these sequences are. These sequences basically are the part that waves their hand and says, hey, this is the part where it becomes meaningful. Yes.
Okay, thank you.
Yep. All right, so we have this elongation, and then as you guys mentioned, eventually we need termination. We need to stop the process. Again, we don't want to just continue to read down till the end of the DNA. So once again, there is going to be a special sequence of DNA nucleotides that tell the RNA polymerase to stop. And this is called the termination sequence. And conveniently enough, that termination sequence is often something like AT, AT, AT. So our RNA polymerase is moving down, making the base pairs, making the copies, and then when it gets to the end, it releases, our RNA moves away, and our DNA goes back together and is completely unchanged by the process. Now, I mentioned transcription is a three-step process, but there is one more thing that has to occur. Before the single strand of RNA nucleotides can leave the nucleus, it must be modified. Basically, what's going to happen to it is that pieces will be cut out. It'll be put back together. So for instance, this section here may be taken out. This part here at the end may be taken out. This part here may be cut off. And so then all of those would then be put together. A, U, G, C, A, C, U, G, C, A. And then once that process is done, we now have what is known as messenger RNA. Oops, that shouldn't have been here. Hold on. Which, once you write messenger RNA once in the exam, on an exam essay question, you can then abbreviate it mRNA. And so that single strand of mRNA now leaves the nucleus. And that is the end of transcription. I've made a bit of a mess of it on the board, so let's look at this with the pretty pictures and the pretty words from your textbook. Again, we are going to use our special enzyme, well, I'm sorry, was there a question? Okay, sorry, I thought someone was speaking. So we need to use our special enzyme, RNA polymerase. Again, it's going to unwind and copy the DNA. But we need to find that special sequence where it's going to be able to begin, like that AAATTT. The fancy name for that special sequence is called the promoter region. So like I said, on that template strand, it may be something as simple as AAATTT. We have that promoter region. That tells the RNA polymerase, this is where you begin. Bind here, start here. Notice we have our template strand, which is the one that contains the information we want. And then, as we mentioned, we have our complementary strand, which is sometimes also referred to as the coding strand. You may use either term when identifying it. So the first step is finding the right place to start, and that is that initiation. Having that RNA polymerase find that promoter region and bind to that specific location. So it can read the one strand of DNA that has the information that we want, that gene for making a polypeptide on it. Notice, unlike before where we completely separate the strands, we're only separating a small portion of it at a time. So that as that RNA polymerase continues to move down the DNA is unchanged by the process. And again, it's going to make a copy of RNA using the RNA base pair rules. So what that means is anytime it sees a C, what is it going to put?
G.
Anytime it sees a G, what is it going to put?
C.
Anytime it sees a T, what is it going to put?
A.
And anytime it sees an A, what is it going to put?
U.
A U. Excellent. Absolutely. So it's using that uracil anywhere it would normally put a thymine. Notice, and here again, because anatomists love their fancy words, during this process of elongation, using this process that we're talking about, we produce this growing strand of, again, your book refers to it already as RNA transcript, but again, it's not truly RNA, a messenger RNA yet. We need to modify it, remember, but we're making this strand, and again, the DNA is unchanged from it. Notice though, at this location right here, we have something interesting going on. We have one strand of DNA being used to make one strand of RNA. And as we know, anatomists love to name everything. So basically, this area where the work is being done, they give a fancy name to it. They call this region the DNA RNA hybrid. Now, I appreciate this is a very simplistic illustration, but your textbook actually has a really, really nice example of this. Notice here, we see this is the region at the start of the RNA polymerase, where the DNA is being unwound. Here at the back is where it is rewinding the DNA, and the DNA is unchanged in the process. So our our RNA polymerase is going this way down the DNA. And notice here's our template strand. This is the one where we're using the base pairs to make a new strand of messenger RNA. And notice they've tried to point out to us that DNA, they've made the backbone purple, because remember, it's made up of sugar and phosphate, but in this case, the sugar is deoxyribose. Whereas here, they've made the backbone blue to remind us that our sugar and phosphate backbone is made of phosphates and ribose sugar. So different sugars, but also using the uracil instead of thymine. And so this working region right here, this area where we're getting the mixing of the two together, not surprisingly, is called the DNA RNA hybrid region. Just like we did, this RNA polymerase will continue down the DNA, making thousands of base pair, making this big, huge strand of RNA until it reaches that endpoint. Like we mentioned, it could be something as simple as AT, that termination sequence. And once it comes off that once it hits that termination sequence, the RNA polymerase releases from the DNA. And again, can't emphasize this enough. The DNA is completely unchanged by this process. But what we do have now is a new temporary and portable copy of the information. This temporary portable copy of the information needs to be modified. It needs to be cut and added back together. And once that is done, then the end result is that messenger RNA. And once we have that messenger RNA, the messenger RNA leaves the nucleus. And how could it leave the nucleus? How does it get out of the nucleus? That nucleus is that big, huge double membrane structure. How the heck does it get out?
Through the nuclear pores.
Yeah, exactly. Perfect. Excellent. Questions on this? So there you go. That is transcription 3. Let's change that. 3.5. Right, because we have initiation, elongation, termination, and then we have to modify the RNA at the end to make our messenger RNA. So it's really kind of 3.5 steps. I'm not sure we need to call that last one a 4th step all by itself. Although if you want to do that because it's easier for you to remember, that's fine as well. All right, questions on this? If this doesn't make sense, it's only going to get worse from here. So if you have questions, ask them now. All right, this is going to then lead to translation. But there are some issues we have to talk about first before we can talk about the process of translation. We will talk about the process of translation, but first we need to talk a little bit about the process and then the tools we're going to need to use to do it. Our first problem is a problem of language. As we've talked about, translation is the process of changing the information from the language of nucleic acids to the language of proteins. Wow, this is way big. Doesn't need to be that big. And here's the main problem. It's something as simple as the alphabet. For our language, how many letters are in the alphabet?
26.
Yeah, 26 letters that we use to combine into words to make information that is meaningful. All right, so for the English alphabet, and that's a more accurate way of saying it, 26 letters in the alphabet. What about the language of RNA? How many letters are in the language of RNA?
4.
4, right? We have A's, we have C's, we have G's, and we have U's. And we have to convert that to the language of proteins. What is the language of proteins?
Yeah, amino acids.
Exactly. And how many amino acids do we have?
4.
Is it 4?
Oh, 5.
Is it 5? How many amino acids are there? Nope,
Yeah.
not 4 or
Oh.
5, but it's also not a gazillion. I told you didn't have to know them, but you need to know how many of them we have.
10.
Close, double that.
20.
There we go. See how easy that was? We have 20 amino acids that are used to make up our proteins. And that's a problem. So if we only have 4 nucleotides, but we have 20 amino acids, does that mean that A has to stand for 5 different amino acids and we get to randomly pick which one we put into the sequence?
No.
No, that wouldn't work, because we talked about, like with sickle cell anemia, just changing one amino acid in a sequence can have devastating effects. So how can we convert the language of RNA into the language of proteins?
Use multiple letters to stand for different
Exactly.
amino acids.
Exactly. We need to use multiple letters, right? So luckily for us, we can do 4 to the 2nd power. Where's my, there it is. 4 to the 2nd power. So that would be stuff like AA or AU or CG or something like that. And if we did that 4 to the 2nd power, how many combinations is that? How many possible combinations? What's 4 to the 2nd power?
16.
16. Is that enough?
No.
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 So notice what we need to do is we need to use a combination of 3. 4 to the 3rd power, something like AAA or AUA or CGA or something along those lines. Notice if we use that, what's 4 to the 3rd power?
64.
64. Excellent. It's 64. That
Yeah.
gives us more than enough. So what we need to do with that RNA is, with the RNA, we need to use sequences of 3 nucleotides to code for 1 amino acid. And we have a fancy name with this, where I saw somebody wrote it, but I missed.
Codon.
Exactly. The fancy term we use for this is a codon. Exactly. So notice if we go back to our lecture here and we talk about our codons, notice any textbook worth their salt at some point is going to have a chart like this. Notice, for instance, we can start something simple. If you have your first nucleotide is, oh, red doesn't show up at all. Let's see if black shows up. First one is C, second one is C, third one is U. It codes for the amino acid proline. Now notice with 64, we have more than enough for the 20. So not surprisingly, there can be multiples that code for the same amino acid. Now, I already told you you don't have to memorize all 20 amino acids. So do you think you have to memorize this chart?
No,
No.
No.
just how to use it.
Yes. Do you need to know how to use it? Yes, absolutely. However, I do notice one other thing. The other thing I notice is there happen to be 4 codons that are highlighted. And if they're highlighted, that probably means they're important. And do you need to memorize the 4 highlighted codons?
Most likely.
Not most likely. How about definitely? Yes, absolutely. You need to know all 4 highlighted codons and what they do. Let's start simple. One of them happens to be highlighted in green. And what does green usually mean?
RNA polymerase.
I'm sorry?
Go.
Yeah, green means go. Green means go, absolutely. And so AUG happens to be what is known as the start codon that tells us where to start the process of translation. However, it also codes for an amino acid. And the amino acid it codes for is methionine. And that one you do need to know. Since we need to know what AUG knows, you do need to know that it codes for the amino acid methionine, as well as being the start codon. Notice there are 3 other highlighted ones, and those happen to be highlighted in red. And what does red usually mean?
Stop.
Stop. These are the stop codons. They're the ones that basically tell translation to stop. Notice also, do these happen to code for amino acids as well?
Yes.
Really, I don't see any amino acids listed there. Notice this one, it actually says methionine, and really it should say start. Do we see any additional amino acids for this one?
No.
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 Notice our start codons do not code for an amino acid. And we'll see why in a bit. So there you go. You don't have to have this chart memorized. You need to know how to use this chart. And you need to memorize these 4 highlighted codons and understand what they do. All right.
So why are there 3 different types of stop translations?
Because, again, why, I don't mean to be flippant, but why is one of those questions that's really hard to answer in biology. And I don't have a good reason for it. It just, that's the way it is. So often we can kind of guess or infer why things are the way that they are. But as I've said many times, the engineer who designed all this did not leave us her footnotes. So we don't know why. We can guess sometimes, and I don't have a good guess for you here. Maybe just because stopping is really important. But then why not have more than one start? I don't know. Maybe because we want to be more precise. Again, totally guessing. No idea. All right. So we understand this. We understand that messenger RNA is going to be needed for translation. We're going to take that messenger RNA and convert it into a protein. But there are two other major components that are needed for this process. Turns out messenger RNA is not the only RNA involved. There are two other very important RNAs that are needed for this process. The first is what is known as transfer RNA, which after you've written it out once, you can abbreviate tRNA. And the other is ribosomal RNA, which after you've written out once, you can abbreviate rRNA. Let's talk about transfer RNA first. Transfer RNA is by far the smallest of our RNAs. A single strand of RNA of only about 80 nucleotides. Now, what's special about this, again, if you think about it, when we were talking about proteins before, we were talking about how there is a primary structure, which is the sequence. We also talked about how there's a secondary structures, which give the individual. Secondary structures where we get the curvatures and things like that. And then that gives us our tertiary structure. The three-dimensional shape. The secondary structure of our transfer RNA kind of looks like a clover. And on this clover, there are two very important parts. Up here at the top is a special sequence of nucleotides that can bind a specific amino acid. So up here at the top, there's a special sequence of nucleotides that combine to a specific amino acid. At the other end, the top of the clover, there is a specific sequence of three, let's make this green, nucleotides that allow it to bind to messenger RNA. Now, messenger RNA contains the information in the form of codons, right? So this special sequence that allows it to bind to the codon is what is known as the anticodon. Let's think really simply about this. What is the very first codon we learned about on our messenger RNA? Come on.
The promoter?
No, again, remember for the codons, what was the first codon that we started with called again?
The
Methionine. Methionine.
methionine.
So methionine is the amino acid it's going to code for. But what did we call that special sequence of three nucleotides?
A codon.
Yeah, which codon was it?
AUG. Start codon?
The start codon, exactly. It was the start codon. And you guys are correct. What was the start codon?
Methionine.
Well, that's the amino acid it binds for, but
Oh my
what
gosh.
was the start codon? Someone said it already. Say it again. There we go. Crystal wrote it out. AUG.
AUG.
So on our messenger RNA, there is a start codon, oops, AUG. So if this transfer RNA is going to bind to our start codon, what would the anticodon need to be? What would the 3 sequence of nucleotides that would be complementary to this, what would it have to be? Well, what's complementary to A? But remember, we're on RNA now, so would it be T? There we go, UAC. So here at the end would be the specific anticodon that would allow it to bind with the codon on our messenger RNA. So that this and only this transfer RNA would be able to bind to the start codon. And as you guys have pointed out to me a couple times, what amino acid would be bound here at the other end of this particular transfer RNA? What is the start codon code for? Now you can say it, Amanda.
Methionine.
There we go. So at this end, it would hold the methionine. At the other end, it would have the anticodon that would allow it to bind to the start codon. So the key to this transfer RNA is it has 2 important binding sites. The first is for a specific amino acid. And the second is an anticodon that allows it to bind to a specific messenger RNA codon. Now, I'm not even going to attempt to draw the 3-dimensional structure of this. And again, you don't need to know it anyway, but for simplicity, we often just draw this as a rectangle, sometimes a bent rectangle, but I'm not going to worry about that, where we can put a particular amino acid at one end. And again, as we mentioned, this particular amino acid would be methionine. And at the other end of the cylinder, we would have the specific anticodon, UAC. So it's often drawn just symbolically like this. So this is our transfer RNA. Again, I've done a truly amazing job of drawing this on the board, but let's actually look what it actually looks like. Again, our transfer RNA has a specific amino acid at one end and that specific anticodon at the other. So here are the pretty pictures. Notice here is the secondary structure, as I mentioned. Here is up at the top where it attaches to the amino acid, and these 3 right here are their anticodon. And just out of curiosity, to make sure we understand this, what codon would this particular transfer RNA bind to? If its anticodon is AAG,
So be C.
there you go. Exactly. See how the game works? Whatever that anticodon is, using the complementary base pair rules, we can figure out what the codon would be. Now, as I mentioned, here is the actual 3-dimensional structure of it, and you can see why I wasn't going to bother drawing that. And as I mentioned, it is often just symbolically shown as a cylinder. And like I said, normally the cylinder, sometimes the cylinder is shown as bent. I'm not going to bother showing it bent. We'll just draw a box with a wick at the top where it attaches to the amino acid and it has the anticodons on the bottom. All right. Questions on this? All right, so that is the first structure we need for, or second structure we need for translation. Now we have messenger RNA. Now we have our transfer RNA. It's happening in the cytoplasm, and that actually brings us to our third component. So that's actually an excellent question, Mitch, because the third thing we need for this process is your friend and mine, the ribosome. Ribosomes, remember, as we talked about, are the workers that make the sequences of amino acids, right? We said they make proteins. Well, really, they make the sequences of amino acids. Ribosomes are made up of two things. The two things they're made up of are proteins, because of course they are, and also ribosomal RNA. So ribosomal RNA and proteins together make our ribosomes. Ribosomes have two subunits. And those two subunits have incredibly fancy and difficult names, small and large. The small subunit is small in size, hence the name. And it has one binding site. That one binding site is for messenger RNA. Oops, RNA. So let's put a little block on this right here. And this little block right here is the binding site for the messenger RNA. So if the small subunit is small, guess what the large subunit is?
Large.
There we go. And the large subunit has 2.5 binding sites. I'm going to cheat with my drawing here. So that one there. That one there. That one there. These three binding sites, again, have very fancy names to them.
Substrates?
Nope, they are the E site. They are the P site. And they are the A site. All three of these sites are for transfer RNAs. A little too small. A site is where the new amino acid comes in. Hence the name A site. The P site is where we find the growing polypeptide strand is held. hence the term P site. And anyone have any idea what the E site is for?
To exit.
Exactly. It's where the transfer RNA exits. And so now we see why they have their names. A site is where we get the amino acid enters. P site, where we store and hold the polypeptides, and the E site for the exit. And so that little globular structure that we've been calling a ribosome, this is basically what it is. It's made up of these two subunits. And again, if you don't like my drawings, here's the pretty picture from your textbook. Again, ribosomal RNA is made up of proteins. Ribosomes are made up of ribosomal RNA and proteins. There's the large and subunit. And their job is to bring the transfer RNA and the messenger RNA together. We do that by having those 3.5 binding sites. On our small subunit, that doesn't show up at all. On our small subunit, we have our messenger RNA binding site. And on our large subunit, we have the A, P, and E sites, the amino acid site, the polypeptide site, and the exit site. So as you can see, Mitch, to answer your question, this is all going to take place in the cytoplasm, basically at the rough endoplasmic reticulum, because that's where all the ribosomes are. Well, not all, or most of the ribosomes. Most of the ribosomes are bound to the cisternae of the rough endoplasmic reticulum. So translation primarily occurs at the rough endoplasmic reticulum. All right, questions on this? All right, we have the pieces, we have the players. Oh, I'm sorry, was there a question? All right, we have the pieces, we have the players. Now the goal is to put them together and see how they work. I said there's a little bit more effort that goes into this, so we will, again, one of these longer elaborate processes, so we'll go through it twice, once on the board and then once with all the pretty pictures and the pretty words. But let's let this information sink in and we will come at this fresh. So this is a perfect place for our first break. So let's go ahead and take our first break now. It is 9:23, so we will restart at, it'll be 9:39. And I will start the recording at that time. All right, any questions? All right, caffeinate, go to the bathroom, drink alcohol, cry, do whatever it is you need to do so that you are prepared and ready to go when we come back. All right, see you guys in 15 minutes. All righty. Let's talk about the process of translation. Any questions before we get started? Excellent, stunned silence. All right, as we know, this process is going to begin when our messenger RNA leaves the nucleus and enters into the cytoplasm, where, again, remember right next to the nuclear envelope is where our rough endoplasmic reticulum is. So it's going to go to the rough endoplasmic reticulum where our ribosomes are set up. And so what's going to happen is our ribosome is going to bind to our messenger RNA. And as we talked about, there are two main components to our ribosome, the small subunit and the large subunit. And which of those two has a binding site for the messenger RNA?
The small.
The small, excellent. So our small is going to bind to our messenger RNA, but it doesn't want to bind at any old random point. where would be a good binding place to, say, begin the process?
The AUG codon.
Why? What's special about AUG?
That's the start codon.
Exactly, and that is exactly what's going to happen. Our small subunit is going to come here and is going to bind to our messenger RNA, as you very clearly and astutely pointed out, at the start codon, AUG. Now, once our small subunit binds, then our large subunit can bind as well. And remember, our large subunit has 3 binding sites. An A site, a P site, and an E site. I'll go ahead and put the letters here now. I don't know how long we'll be able to leave them here, but we'll make it work. E, P, A. Excellent. So now our large and our small subunits of the ribosome have come in and bound. Excellent. And these A, P, and E sites, what are they binding sites for?
Amino acids and the
Well, true. What's going to come is going to bring an amino acid, but what is the binding site actually for?
For transfer RNA.
Exactly. So a transfer, oops, hold on, one dot to stay blue. Exactly. So a transfer RNA is going to also come into this first site where the start codon is. However, can any old transfer RNA come and bind at this location?
no.
No, it has to be a specific one with a specific anticodon. And what did we say the specific anticodon would be?
U AC. ACM.
Perfect. So let's make this the right size. Let's make this the right color. Let's make it fit. Yeah, that is too big. That doesn't look right either. That's weird. Why did I do that? There we go. Excellent. So the right anticodon. And what amino acid did we say that this would bring with it?
The methionine.
Methionine. Excellent.
Methionine.
And again, I don't care how you pronounce. I don't care how you pronounce these things as long as you spell them correctly on the exam. So it brings our first amino acid, and we happen to know in this case that this amino acid happens to be methionine. So we'll go ahead, oops, and put met in there. Excellent. So let's talk about what has happened. At the beginning of translation, if you notice, 4 things are coming together. Our messenger RNA, the small subunit of the ribosome, the large subunit of the ribosome, and our transfer RNA. So those 4 things all come together, and they all come together on that start codon. So basically it read down the messenger RNA till it got to our start codon, and then all 4 of these things came together. All right, we comfortable with our starting point? Excellent.
Yes.
Now notice we have an A site, and someone remind me what A stands for again?
Amino acids.
Amino acids. So notice we have a spot to bring in a new amino acid. So a new transfer RNA could come into this site. Again, can it be any old transfer RNA?
No.
No, it has to be a specific transfer RNA with the specific anticodon. And what would that anticodon be in this case?
It would be
There we go. Crystal's got it.
GCU.
GCU. Excellent. So only the correct transfer RNA with the correct anticodon could come in, and it will bring our second amino acid. Now, just off the top of your head, do you happen to know what CGA codes for as far as an amino acid?
No.
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 Right? If we cared enough, could we go to the chart and look?
Yes.
Yeah, but we don't really care. Does it really matter? No, not really. So we'll just call it amino acid 2 because it really doesn't matter. So we know amino acid 2 with the specific one, whatever it is going to come in and bind in this site as well. All right. Questions on this? All right. Now that we have both our A and P sites, oops, hold on, I don't want this to be capped anymore. And actually, I'll just abbreviate it. Now, two chemical reactions are going to take place. All right. Made me to make this smaller to make it all fit. So two chemical reactions are going to take place. The first thing we are going to do is we are going to make a bond. What kind of chemical reaction did we learn about is used to make a bond?
A covalent bond?
Yeah, if we're going to form a covalent bond, putting these two things together, what was that synthesis process that we learned about where we put them together? Remember, during that process, we pulled a water out of it.
Hydrolysis?
Well, remember, hydrolysis is how we use water to split a bond. In this case, there we go. Dehydration synthesis. Absolutely. So we are going to use dehydration synthesis, and we're going to use that to form a bond between the two amino acids. Now, if we're forming a bond using dehydration synthesis, then as a result of that, we are going to pull out a water. All right, water is going to be drawn out in this process. And we can use the water for our second chemical reaction, which somebody already mentioned. We want to break a bond. And what type of chemical reaction are we going to use to break the bond?
Hydrolysis.
There we go. We're going to use this water, and we use the water to break the bond between our first amino acid and the transfer RNA in the P slot. So two chemical reactions take place. First, we make a bond using dehydration synthesis. We bind the amino acids in the P site and A site together. And then we break a bond. And we use hydrolysis to break the bond between the amino acid and the transfer RNA in the P site. So notice two chemical reactions took place. We made a bond and then we broke a bond. Questions on that? All right. At this point, there's nothing else that our ribosome can do at this location. So think about yourself. Friday's right around the corner, and there you are like me, sitting on Grindr, looking at all those pretty pictures. And when you see something you like, what do?
Double tap.
True, or you
What?
swipe right. You swipe right, right? Well, that's exactly what happens here. After these two chemical reactions take place, our ribosome shifts to the right. Notice as it shifts to the right, and I got to cheat and do it 1 unit at a time. Notice as this, oops, not you. So that goes there. So notice what happens when this shifts to the right. The transfer RNA that was in the A site is now in the P site, holding the strands of the polypeptide. And this first transfer RNA is now in the E site. And what do things in the E site do again?
Exit.
Exit. And that's exactly what happens. Whoops. It exits, going back into the cytoplasm, where eventually it might run into an enzyme that puts another amino acid on the top of it, but we don't really care what happens to it at this point. It's gone, we no longer care about it. However, what we do care about is that there's an open A site. And guess what happens in that open A site?
It needs to get a new codon.
Exactly, a new, well, not a new codon, but you got the right idea. We need to bring in a new amino acid. And what's going to bring that new amino acid in?
tRNA.
A transfer RNA. Can any old transfer RNA come into this site?
No, specific to the mRNA.
Excellent. And what would the anticodon be here?
G.
Now you skipped one. We're starting with the
Oh.
G actually.
Oh, CAA.
Yep. Excellent. So with the correct anticodon, and again, I appreciate my drawing's not the best, so that's why I can understand the confusion, is going to bring in the next amino acid. Do we happen to have that one memorized?
No.
Could we figure it out if we cared?
Yes.
Do we care really right now?
No.
No. We just know it's going to be the third amino acid. Oops. And again, if we needed to find out, we could grab a table and we could figure it out. But for this purpose, we don't care. Excellent. Now all my transfer RNAs are locked and loaded. So what's going to happen next?
The chemical reactions.
Yep. Which one happens first?
The dehydration synthesis.
Excellent. So we make a bond. Where do we make that bond?
Let's see.
Anyone, where do we make the bond?
Between the two amino acids.
Exactly. We make that bond between the two amino acids. And you're 100% correct in that process. We end up drawing out a water because it is a dehydration synthesis. So we get that water. And then what do we use that water for?
To break the bond.
And what?
Hydrolysis.
Yep. And where are we breaking that bond?
Between the amino acid and the tRNA P site.
Excellent. We use it to break the bond between the transfer RNA and the amino acid in the P site. Excellent. We did our two chemical reactions. We made a bond and we broke a bond. So what do we do next?
And swipe right.
Swipe right. Excellent. Notice as we shift to the right, our second transfer RNA is now in the E site. And what does it do?
Exits.
It's going to exit. Our A is now in the P site. Oops, didn't shift my small subunit. So what happens next?
Another amino acid enters the A site.
True, although we want to be careful with our verbiage. Yes, an amino acid comes in, but what brings it?
The tRNA.
Right, so a new transfer RNA enters into the A site. Remember, the A site is for the transfer RNAs. Again, any old transfer RNA?
No, specific to the mRNA code.
Excellent. And what would that specific anticodon be here?
That would be GGZ.
Excellent. Oops, didn't make them big. Excellent. And again, it is going to bring our fourth amino acid. And again, off the top of our heads, we don't happen to know what it is, but as we mentioned, it doesn't quite matter right now. We just know that the next one's coming in. Again, hopefully you are noticing and appreciating that it is the same pattern repeating over and over again. So hopefully these questions are getting easier each time. Excellent. We are now locked and loaded with two amino acids here. So what happens next?
The chemical reactions
Yep.
between the amino acid 4 and 3.
Excellent.
The
So
dehydration synthesis using the hydrolysis.
excellent. Well, careful.
Oh,
Dehydration, we make a bond using
right.
dehydration synthesis. Absolutely. And as a result of that dehydration synthesis, we get a water. And then what is that water used for?
To break that bond using hydrolysis.
Exactly. We use it to break the bond between the transfer RNA and the amino acid in the P site. And now that both of our chemical reactions are complete, what's going to happen next?
We're going to swipe.
Exactly. It shifts to the right. And notice, this is going to happen hundreds, if not thousands of times. Remember, a single polypeptide is made up of hundreds and thousands of amino acids. So this is going to happen 100 or thousands of times.
Does this happen pretty quickly?
Yes. So we're shifting to the right. The transfer RNAs are coming in, make a bond, break a bond, shift to the right. Add a new transfer RNA, make a bond, break a bond, shift to the right. And this process continues again and again and again and again and again and again until something special happens. What's that something special that's going to happen?
It's going to be the stop translation.
Well, stop codon.
Oh, stop codon, sorry.
And what are the stop codons?
It's UAA, UAG, and UGA.
Oh, and conveniently enough, what have we run into here?
UGA.
We have indeed run into UGA, and isn't that one of our stop codons?
Yes.
Excellent. So now that we're at a stop codon, we're going to see something slightly different happens. Again, a transfer RNA is going to come in. And again, can it be any old transfer RNA?
No.
No, it has to be one specific to the codon. So what would our anticodon here be?
GCU.
GCU?
Oh, am I off again?
UGA.
Oh, sorry.
There we go. A. C. U. Excellent. So just like before, a transfer RNA with the correct anticodon is going to come in. But if you remember, unlike the start codon, which codes for an amino acid, methionine, remember we said the stop codons don't code for an amino acid. And that's because our stop codons, our stop transfer RNAs don't bring amino acids. It turns out what they bring is a water. And what might this water be useful for?
Break the bond.
Exactly. This water is going to be used for hydrolysis to break this bond. And notice, once we break this bond, is anything being held together anymore? Is there an amino acid here now anchoring this polypeptide in place? No. So once this hydrolysis takes place, the whole thing comes apart. Our large and our small subunits come off. The rest of our transfer RNAs go away. And what we are left with after this process is now a sequence of amino acids. And this sequence of amino acids now travels to the Golgi apparatus.
Is it always a sequence of 4?
No, remember, as I mentioned, it's hundreds or thousands long.
Oh, okay.
I just wasn't going to do it hundreds and thousands of times.
Okay.
So let's actually put that on there to remind ourselves. Hundreds to thousands of amino acids. And this sequence of hundreds of thousands of amino acids then travels to the Golgi apparatus to be assembled. Into a functional protein. Excellent. And there you go. Just that simply, just that easily, we have described the process of translation. Questions on that?
One quick question. So when the codons exit, is there anything holding on to those, or a new what's it called? Does a new tRNA find it and recycle it?
So, okay, great question. You have the right idea. So again, remember, these codons are not free floating by themselves. Remember, these codons are held together in a single strand of messenger RNA. Okay. So when it comes apart, the messenger RNA, notice, is unchanged by the process. So is there anything stopping an additional ribosome from binding onto the start codon and beginning the process?
No.
No. In fact, if you think about it, once that first ribosome starts moving down and out of the way, another one can actually bind on before it even ends. So that single messenger RNA can be read hundreds of times. Which, if you think about it, makes sense. If our cell needs to make a particular protein, do you think it just wants one copy of that protein?
No.
No, probably not. We probably need hundreds, if not thousands, of those proteins. And so a messenger RNA can be read hundreds, if not more times, to be used to make a lot of proteins. All right. Questions on that? All right, we've done it on the board. Let's look at the pretty pictures from your textbook and go through the whole process again. I'm going to start with this really simplistic illustration, but I will point out your book has an in focus that does an amazing job of showing this. Notice with this simple process for the large subunit, they don't even bother showing the E site. They just show the binding sites. But as we can see, all four parts, the messenger RNA, the small subunit, the large subunit, and our first transfer RNA all come together on that start codon. First, the small subunit binds to it, then the large subunit, then the first transfer RNA. And what's cool is, since we know the start codon is always UAG, we know the anticodon, the first transfer RNA, is always going to be UAC. And we also know what amino acid it's going to bring. It always brings that methionine. So all four of these things all come together always on that start codon. All right, questions on that? All right, once our first transfer RNA comes in, what has to happen next?
The chemical reaction.
Well, before we have a chemical reaction, what do we need? Don't we need a second amino acid? Yeah, we need a second transfer RNA to come in.
Oh, yeah.
Come into the A site. Again, it can't be any old transfer RNA. It's got to be the one that has the specific anticodon to the codon on our messenger RNA, bringing in our second amino acid. Once it's in there locked and loaded, it's time for our chemical reactions. And how many chemical reactions are there going to be?
2.
Excellent. What happens first?
The dehydration
Synthesis.
synthesis.
Excellent. Where we form a bond between the two amino acids. And as a result of that, we get a little bit of water. And then what do we use that water for?
To break the bond.
Right, for hydrolysis,
Hydrolysis.
to break that bond between the first amino acid and the first transfer RNA. Once that occurs, what happens?
It shifts to the right.
Exactly, swipe right. Right, and as that occurs, our first transfer RNA leaves. Our second one is now in the P site. And our A site is now open for the next transfer RNA to come in. And again, this process continues hundreds, if not thousands of times, until finally something special happens. What's that something special?
The stop codon.
And what are the 3 stop codons?
UAA, UAG, and UGA.
Any of those three. When we reach that stop codon, as you can see here on the pretty picture, here's our next codon. It happens to be one of the start codons. And remember, at that point, as we mentioned, our transfer RNAs, oops, brings a water. That water is used for hydrolysis of that last bound amino acid, at which point everything comes apart. We have our completed polypeptide, the large and subunits come off. And again, our messenger RNA is not changed by this process, so it can be read dozens of times. Your book's got this couple great pictures. So again, we had a very simple illustration. This is the in focus from your textbook, which does an amazing job of describing this process. If the way I've described it has been challenging and doesn't make sense, then I encourage you strongly to read this where they are talking about this in your textbook. In fact, notice they also show how the transfer RNAs have a special enzyme that puts the specific amino acid on it. You don't need to know this process for the exam, but it's a cool process that it explains and everything that goes along with that. And then the other thing that your textbook has that I really like is, remember, as I mentioned, a single strand of messenger RNA can be read by multiple ribosomes at the same time. Here is an illustration from your textbook showing this, but what your textbook actually has that's really cool is actually this electron microscopy view, where we can see, using an electron microscope, a single strand of messenger RNA that has about a dozen ribosomes on it, all reading it, all making amino acid sequences at the same time.
Crazy.
Yep, the body's cool. All right, questions on this?
And that comes out of the nuclear pores, right? The mRNA?
The messenger RNA comes out the nuclear pores to the cytoplasm. Remember, the ribosomes are in the cytoplasm. So notice, this here is the exact process we just described, this process of protein synthesis. We started with a gene on one single strand of our DNA. Here is that strand. Notice we were able to convert that from DNA into RNA. And that RNA, which is our messenger RNA, has the codons on it. Those codons correspond to the anticodons on our transfer RNA, which are used to make a sequence of amino acids. You absolutely positively need to be able to describe this process. And if I gave you a chart for which amino acids went with which anticodons or which codons, could you actually give me the sequence of amino acids?
Yeah.
If I gave you a strand of DNA, could you give me the sequence of amino acids that produced it? If I gave you a chart.
Yes.
Yes. And in fact, if I gave you a chart, could you also do it the other way as well? If I gave you a sequence of amino acids, could you give me the DNA that would produce it?
Yes.
Yeah. So are those two things that would make good essay questions on the exam?
Yeah.
Yeah, absolutely. All right. One more important question. Well, we kind of just did. But before we do that, I do have one more important question that I want to ask, and then we'll answer your question, Augustina, and then we have one other piece of information we need to talk about. Here's the problem that we have. Your toenail cells, your eyeball cells, your liver cells, do they make the same proteins or do they make different proteins?
Different proteins?
Different proteins. Does that mean that your toenail cell, your eyeball cell, and your liver cell have different DNA?
No.
No, the DNA is the same in every single cell in your body. So how is it that our toenail cell can make toenail proteins, our liver cell can make liver cell proteins, and our eyeball cell can make eyeball proteins. If they all have the same DNA, and remember, as we talked about, only 1.5% of our DNA is even proteins, codes for genes anyway, how is it possible that I can tell? This is kind of goes back to the question Augustina was asking us before. How in the world do I not only tell my toenail cell which proteins to make, but when to make them?
Is it enzyme specific?
Close. You've got the right idea. It does have to do with a chemical, but in this case, the chemical isn't an enzyme. How do you think we regulate this process? What organ systems do we have that are involved in regulation or communication in the body?
Our nervous system?
Nervous system. So does that mean that every single cell in our body has a nerve going to it, telling it which proteins to make?
Yeah, possibly.
True, really? Every single cell, every single one of your
no,
skin cells has a nerve going to it?
no.
So what was the other organ system that was specialized for communication?
The nucleus?
Organ system. Endocrine, absolutely.
Endocrine.
So the primary way we tell cells what proteins to make and when to make them are hormones. We use those hormones to be able to tell cells what proteins to make and when to make them. And that's how we get toenail cells to make toenail cells proteins, eyeball cells to make eyeball cell proteins, and liver cells to make liver proteins. We give them hormones that tell them what to do and when to do it. All right. And again, I think it was Augustina that asked it before. Notice here, this right here is a perfect example of that. If you had a chart, I could give you either of these two pieces of information. I could give you this sequence of DNA, and showing your work, I could ask you to give me the sequence of amino acids. Or I could do it the other way as well. I could give you this specific sequence of amino acids and ask you to give me a strand of DNA. The only thing I would have to do is I would have to give you that codon chart or an anticodon chart. If I gave you one of those things, because you don't have to memorize which codon or which anticodon codes for a specific amino acid, but as long as I gave you one of those two charts, you should be able to do this in either direction. All right, questions on that? I have one, go ahead.
Oh, I said no.
Okay.
Sorry.
I have one other very important piece of information to ask you. Obviously, we've spent all of today talking about protein synthesis. And protein synthesis is made up of two related processes. So being the smart, sophisticated students that you are, how many possible essay questions could I give you based on this? Oops, why is that not right?
Four.
4. I was thinking more along the lines of 3. All right. So obviously, question 1 is describe process A. Question 2 is describe process B. What's the third possible question?
Compare.
Exactly. Right? And that could be things that are similar or things that are different. Now, if I were to ask you essay question 3, does that mean describe all of process A and describe all of process B?
No, just how they're similar
No,
or different.
Right. So on the exam, if I told you to compare the processes of translation and transcription, and you described all of translation and all of transcription, and all of your information was 100% correct, would you get 100% of the points?
No.
No. Excellent. Now, as I've said many times, format isn't important. What I care about is the information. However, for these types of comparison questions, I sometimes for myself find it very useful to think of it in terms of a table. Because when we do a comparison, we want to make sure we are comparing apples to apples and oranges to oranges. So the way I do this is actually set up a 3-column table where I have transcription
Throughout
here,
the end.
here, and translation here. And then over here in my 3rd column, I'm going to put the characteristic that I'm comparing. So let's do this. Let's pretend this was the essay question you got on the exam. And I asked you to compare translation and transcription. Tell me some ways they are different from each other. Give me an example of a way these two processes are different.
Transcription is DNA to mRNA, and translation is nucleic acids to amino acids.
Okay, so one way their differences are the languages used. Right? In transcription, you're right. We go from nucleic acids to nucleic acids, so it doesn't really change. Or it changes a little bit, right? We're going from DNA to RNA. But that's vastly different from what's happening in translation. In translation, you're right. We are going from nucleic acids to amino acids, right? Dramatically changing the language there. Excellent. I like that. What's another way they are different?
Transcription is the unwinds the DNA.
Does translation do anything to the DNA?
Well, yeah, also copies, but it
Does it
unchanged.
copy the DNA?
No.
No. So again, we want a direct comparison, right? Mitch has got a good one. How about locations? Location where they occur. Where does transcription occur? Nucleus. And where does translation occur?
Cytoplasm or ribosome.
Well, cytoplasm is not an incorrect answer. It's a little bit generic. We can be more specific, like the rough endoplasmic reticulum.
Okay.
Or if we wanted to be really specific, as you guys pointed out, specifically at the ribosome. Because remember, most ribosomes are bound to the rough endoplasmic reticulum, but some of them are free floating. So does it 100% have to happen at the rough endoplasmic reticulum? No, but it 100% has to happen at a ribosome. So any of those are really acceptable answers in this case. I like where you were going before, but I think your description was a little off. If you instead think about the information that is being read, right? Transcription, you are correct, starts with the DNA, and that's where we're getting the information. What are we reading in translation?
The codons.
True, but those codons are the messenger RNA. Excellent, right? Right. And again, don't be using abbreviations like all of this. On a real exam, you'll be writing them out. And then if you want to flip it to the same side, what's the end product of these processes? At the end of transcription, what do you have?
You have mRNA.
And after translation, what do you have?
Don't you still have mRNA?
Not at the end of the process. What do you have at the end of translation?
RNA.
Amino acids.
Yeah, you have amino acids, or let's be more specific, a string of amino acids, or if we want to be more specific, we could say a polypeptide. Right?
Yeah.
Either of those would be acceptable answers. Anybody think of another one next at the top? I can think of one last that just off the top of my head. Proteins involved. What primary protein is involved in transcription?
RNA polymerase.
Yep. Whereas for translation, the proteins plus the ribosomal RNA form the ribosome. So there, the proteins are helping to form the ribosome that is involved in the process, where transcription, it's primarily RNA polymerase. And there's probably a half dozen more we could probably come up with as well. Notice the key to this type of question. I am directly comparing things, right? And again, I want a direct comparison. Saying that transcription starts with the DNA and translation doesn't, is that really a good comparison?
No.
No. Is saying that transcription starts with DNA and translation occurs in the cytoplasm?
No.
Is that a direct comparison? No. So again, like I said, it's not required to set it up this way. But for me, it helps me to organize to make sure that I'm really comparing, right? Oops, that there. Even I've messed it up now. Hold on. So that went there like that. It helps me to know and understand that I'm really comparing, right? Apples to apples. I'm not just describing the two processes. I am actually literally comparing them, directly comparing them.
Are we able to set this up like this, like within our text box?
You can't draw the arrows, but certainly
Right.
within a text box, you can do that. Absolutely.
And that's acceptable for like,
Yeah.
and okay.
Absolutely. Yeah. Like I said, I'm less concerned about the format and more concerned about you having correct information. Correct information, details, specificity. That's the things that I'm looking for. All right. Questions on that? So there you go. 2 related questions, 3 possible essay questions. And again, since this is all we're doing today, are you incredibly likely to see one of these 3 essay questions on the exam?
Yes.
Yeah, absolutely. So is there any reason why you shouldn't, between today and Monday, practice writing this, describing translation, describing transcription, and comparing the 2?
No.
No. And one last question. When I ask for a comparison, I certainly will give you a number, give me 4 differences or something along those lines. So again, make sure you write 4 things. So if I ask for 4 comparisons, make sure you give me 4. If I ask for 5, make sure you give me 5. If I ask for 2, don't give me 4. Because as I said before, if 3 of them are correct and 1 of them is wrong, and I've only asked for 2, what am I supposed to do with that? Can I give you full credit for that?
No.
No, because I'm left trying to figure out, all right, did this person really understand it and just made a small mistake? Or were they trying to throw everything they could think of at the board, hoping that some of it stuck, right? I'm not going to interpret your answers. Your grades are going to be based by what you put on the page. So be very, very careful putting the information on the page. All right. I use this example in class all the time because, and I don't have a good relevant example of it, but before the exam, we may be standing outside of the class, and a student could ask me a question about transcription, and I could ask them to describe it to me, and they can do a perfect job of describing transcription to me. So I know with 100% certainty that they know that information. And then we get in the class and they get the exam. And on the exam, it says, describe the process of transcription. And they accidentally misread the question and describe the process of translation. Now, literally 5 minutes before the exam, I talk to them and I know they know the entire process of transcription correctly. Can I give them credit on what I know they know?
No.
No, I have to do it based on your answers. I have to give you your grades based on what you put on the page. So put details, be specific. And as I've said many times, and I will continue to say, read the questions carefully so you can answer them correctly. I will say this right now in hopes that it doesn't happen. But I say this every semester, and every semester at least one person still makes this mistake. On the exam, I may ask for the process of transcription, and the student describes translation. They picked the harder process, and they described it 100% correct. Clearly, you're showing me you have knowledge and you understand this process. But can I give them credit for that?
No.
No. Every single test, it happens at least once. One person, instead of doing transcription, does translation. Instead of doing translation, does transcription. People often lose points, not because they don't know the information, but because they don't read the questions carefully. All right, that is actually a great lead into what I want to do next. And so here, like this would be, for instance, the kind of thing like on the exam, if I gave you something like this, then you should be able to use this chart to be able to complete that activity. All right, that is everything we need to do from a lecture standpoint for today. So actually, let me put this back on the board so I have some place to write. We obviously still have a lot of time left, but as I mentioned, I want to be able to do a review. So here is what we will do. We will take a quick break. The review will start at, we'll say give it 15 minutes, so 10:48. Again, this is optional. If you're done and want to leave, you're welcome to do that. But I expect the review to be question and answer. If there are concepts that you are struggling about, or you want to go over the stuff we did today, or if you want to go way back to the first day of class when we were talking about, organ systems and all that, whatever information you are having challenges with, this is your opportunity to ask those questions so we can work together as a group to make sure we understand these concepts so that you are successful on the exam. So we will take a quick break, come back, and then I'll spend whatever time you guys need answering questions. All right, any questions? All right, I will see you in 15 minutes. So, as I mentioned, we are now to the point where we are going to do a question and answer review. So like I said, I am happy to answer any questions that you guys have to help you to be prepared, whether either it's on the protocols for the exam, how it's going to be run, or any particular topics as well. Augustina, go ahead.
I mainly have two, but the first one that I'm still kind of confused on is the secondary active transport. How I don't get how it's not directly using ATP, but at the same time is using ATP.
Okay, perfect. I think that's a great question. So you are absolutely correct in that when we talk about active transport, we are talking about either the direct or indirect use of ATP. All right, so either you're directly using ATP or indirectly using ATP. The example we used in class, and there are plenty of them, is if we have a particular protein and that particular protein has ATP directly binding to it. So the ATP comes in and binds to it. When it binds to it, what happens is that one of the phosphates is ripped off, and the energy from that allows this molecule to do work. And typically that work is going to be in the range, oops, that should have been ATP. Not sure what happened to P there. That work comes in the form of moving some molecule against its concentration gradient. Like say, for instance, calcium. Cells don't want calcium inside of them, so we need to use energy for the cell to get it out of the cell. So that is the direct use of ATP to do work. All right. Like I said, I think the direct, or again, this therefore is what we would call primary active transport. I think is the simple example. So hopefully that makes some semblance of sense. Are we comfortable with that?
Yeah, that one makes sense. Just the
Perfect. Okay.
secondary that doesn't make
I
sense.
just want to make sure we have our starting point. Excellent. So for the secondary active transport, active transport, the key for this one is that it does not directly use ATP. So again, we are going to have some type of protein that is going to be embedded within the plasma membrane. However, in this case, what this protein allows is it allows something that really wants to get in the cell, like for instance, sodium. It allows that sodium to enter into the cell. Sodium, remember, has a huge driving force. It really, really, really wants to be in the cell because there is a ton of it outside and there's very little inside, because it's positive outside and it's negative inside. So because it's negative in here, because there is few sodium in here, sodium really, really, really wants to come inside. All right, we comfortable with that?
Mm-hmm.
So what this protein does is it says, okay, sodium, you can come in, but I'm going to use the energy of you coming in to help me to do work. And so that could be being used to bring something else in as well. Like for instance, something like an amino acid. We need to get that amino acid into the cell so that we can make proteins. Or we could see another example of this where we have a protein that again allows sodium into the cell, because sodium really, really, really wants to come into the cell. And we can use the energy of the sodium coming in the cell to kick something else out. So for instance, again, we don't like calcium in the cell, so we can use that sodium coming in to kick out a calcium. Okay, notice these two proteins allow two things to move, and so both of them are known as co-transporters because they allow two things to move, with one thing bringing in another. But notice they're moving them in opposite directions, so they also have different names as well. When they, move this out of the way a little bit. When both things move in the same direction, we call it a symporter. And when they move in opposite directions, they're an antiporter. And those are our co-transporters. Again, notice these are using the energy of sodium. Not using the energy of ATP. Notice nowhere, anywhere on this have I drawn an ATP, because they're merely using the energy of sodium to do work. Instead of using the energy of ATP, They're using the fact that sodium really wants to come in to do work. All right, with me so far?
Yeah.
Okay, so now let's add the last piece of this. Here's the problem with this system. More and more sodium is going to be entering into the cell as we continue to get it to do work. Now, sodium is a positive ion. So as that positive ion enters the cell gets more positive. Sodium is coming in because it has a concentration gradient. But as more sodium comes in, there's less of a concentration gradient. And so if sodium keeps coming in and the cell keeps getting more and more positive and the cell keeps getting more and more sodium inside of it, is sodium going to continue to want to come in forever?
No.
No, eventually it's going to want to stop coming in. And if it stops coming in, we can't get it to do work for us anymore. And suddenly our co-transporters are going to stop. The only way this works is if sodium really, really, really wants to come inside. So we let it come inside, but as soon as that sodium gets inside the cell, whoops. As soon as that sodium gets inside the cell, what do we need to do to that sodium? Anyone?
Transport it out.
Yeah, do we want it to stay in the cell?
No.
No. So what we need is we need a special protein that can kick that sodium out of the cell.
Potassium?
Well, no, I understand where you're going with that. We'll talk about that in a second.
Okay.
And for this sodium to be kicked out, what does this new protein need?
ATP.
Energy, because
Yeah,
it's
it needs
going
energy. Exactly.
across
So
its concentration gradient.
Exactly. To get sodium to go outside, we need to use ATP. So notice the co-transporters themselves do not use ATP. But if we don't use ATP to move the sodium out, the co-transporters will stop working. So that's what we mean by indirectly. ATP does not actually bind to the co-transporters themselves. But if the cell doesn't use ATP to kick that sodium out, then it's not going to be able to continue to do work. And so that's why it's an indirect use of ATP. ATP doesn't bind to the co-transporters themselves. But if the cell doesn't use ATP, these co-transporters will stop. Does that help it to make sense?
Yeah, so the ATP only binds to the proteins that eventually kick out the co-transporters.
Two pumps, right? Our sodium is going to be kicked out by pumps or transporters, absolutely. So it doesn't bind to the co-transporters themselves, but there are primary active transporters, which remember the other name we give for these are pumps or ATPases, because they use ATP to move things against their concentration gradient. Oops, where did that write? All right. Does that help to explain what a secondary active transport is and what we mean by indirectly using ATP?
Yeah. So kind of going along with it, in the lab, there was a question, like on the lab server, It was something along like if we don't have either one of potassium or sodium, that the pumps wouldn't work.
Okay, excellent. In that case, and that kind of goes with the other question someone mentioned earlier. There are many types of pumps or ATPases. The most common one found in the cell, and the one that you were doing in that labster, is a very specific pump that is called the sodium potassium pump, or the sodium potassium ATPase. Now, this is a primary active transporter, so it needs to use ATP. The ATP directly binds to it. And it uses the ATP to do two things. What are the two things that this particular pump does?
Isn't it bring in sodium and then kick out
Potassium.
So do we really need to use energy to get sodium into the cell?
No.
So which way, if we're using energy, which way are we going to want to move sodium?
In?
Sodium really wants to come in, so I don't need to use energy for that. So I need to kick sodium out. And someone remind me, how many sodium get kicked out?
2.
Two?
Oh no, 3.
3 sodium out of the cell.
And 2 potassium in.
Exactly. So notice it uses the energy from the ATP to do both this, to move 3 sodium out and 2 potassium in. Now, if you remove all the potassium, will this pump still work?
No, it's specific. It has to have both potassium and sodium.
Yeah, exactly. Are there different pumps that just pump sodium and different pumps that just pump potassium? Yes. In fact, there are some pumps that pump sodium, potassium, and chloride. So there can be some of these pumps that can do, 2 or more things. But this particular one has to have both sodium and potassium to work. So if one of those is missing, then this pump won't work properly. Now, in a real cell, you don't have just this one, this one pump. There are plenty of different types, and so you can use different ones to move those things. But specifically for the sodium potassium pumps, both need to be there. All right. Did that answer all your questions, Augustina?
Yeah, I have another one, but someone else can go.
Okay, well, let me ask the other two and then we'll come back around to you, okay? Any other questions on this active transport before we switch topics?
It's related.
Yeah, then that's why I asked.
Okay. For the lobster, there were 12 different transports and transporters and channels that they showed. Do we need to know all the names of those ones?
It's been a while since I've done that one. I did that this summer, so I don't remember. What I would say is you certainly need to understand the different types. Okay, so the co-transporters versus the pumps or the ATPases or things along those lines. And what I would say is most of them are pretty obvious. I mean, what do you think a calcium pump does?
Pumps calcium.
Yeah, pumps out calcium. The sodium potassium ATPase is probably the tricky one because it pumps both calcium and potassium, sodium and potassium, but 3 sodium and 2 potassium. So I don't remember what specific ones are in there, but definitely know specifically what this one does. And I think, and certainly understand the difference between a symporter and an antiporter. But I think in most cases, I think, a sodium pump or a calcium pump, I think the names are pretty obvious. And again, I guess the other part of that too is you should know the general concentration gradient, so that if I say there is a calcium pump, for instance, you should know which way it's going to move calcium. Or here, since I haven't drawn it here, what about a chloride pump? If there was a chloride pump, which way would it move chloride?
well, it would want to come in,
So
but
then if we were pumping it, which way would we pump it?
out.
Bingo, exactly. All right. So did that make sense, Aubrey? I know I didn't directly answer your question, but I think
Mhm.
I think that hopefully that's, again, the names of, Bob's Pump or whatever it is aren't going to be important. Most of them have, names specifically that tell. I think the only one, like I said, that's tricky really is the sodium potassium because of the numbers involved in it.
Okay, there was just new stuff on it that was concerning I guess light and voltage. I'm trying to remember.
We are going to get to
Oh, the voltage
gated
carrier.
and chemically
Yeah.
gated. Those are things we will get to, but aren't things that you necessarily need to know for this time. You need to know that there are gated channels, things that open and close, but absolutely as we move forward, we will build towards that.
Okay, cool. Thank you.
Excellent. Great question. All right, Amanda, let's answer yours.
Yeah, I had a question about the physio labs. Is there going to be anything similar to that on our exams? Because the only thing I was confused about is how, the timing that each one would, with the concentration, how quickly it reached equilibrium.
So
And then,
go ahead. And then
it'll ask like which one, transferred the most or something, and I was just confused by that.
Okay, so here's what I would answer to that. So the answer to that is twofold. Any activity, especially a graded activity like that, that takes a lot of time, do you think that that's something that could easily relate to an exam?
Yes.
Yes, absolutely. Now, do you need to know the exact timing of what it takes for a sodium channel to allow sodium to move down its concentration gradient and reach equilibrium? No. However, if you had two beakers, for instance, that both had membranes down the center with sodium channels on them, and one side had 75% sodium chloride, and the other side had 15% sodium chloride, and the other one had 90% sodium chloride, and the other side had 80% sodium chloride. While you don't necessarily need to know how long it would take them to reach equilibrium, do you know which one of these would have the more rapid diffusion occurring?
The bottom one.
Why would the bottom one be more rapid?
Because there's less to.
Okay, so I'm not saying about how long it would take to reach equilibrium. Which one would have the more rapid diffusion at the beginning?
The top one.
The top one.
Because it has a greater concentration gradient, right? Or
okay, and it wants to quickly reach.
Yeah, how long it takes to reach equilibrium isn't as important as the driving forces on it. The top one has a higher concentration gradient, so it will move faster, right?
Okay.
Temperature affects it, size affects it, distance affects it. Understand the things that can speed up or slow down diffusion, but the absolute time that it takes for them to reach diffusion, that's not something that's important. They're indicators of what can be faster or slower. But that absolute number isn't something that's important.
Okay.
Okay. All right. Augustina, you have another question?
Yeah, can we go over the chemical versus the electrical force of, what was it? I think it was during our last lecture where it was during the electrical force versus chemical force during diffusion.
Absolutely. Now, what I will warn you about is to not spend too much time focusing on this particular concept yet, because this will be much more important when we get to the muscular system and when we get to the nervous system. So the point of talking about it now was to introduce you to these concepts, but obviously we're not emphasizing these yet. So let's just explain them in very simplistic terms. Basically, all ions, let's start over here, have a driving force. They want to reach equilibrium. right? We're comfortable with that idea?
Yeah.
Excellent. And actually, I'm going to erase my cell here for a minute to go with our beaker because it's a little simpler. As we've talked about earlier, I put this membrane down the center, and let's use the numbers we just finished using. 75% glucose on this side, and 15% glucose on this side. All right, I poke holes in my membrane. Oops, no. And glucose moves. Which way does glucose move?
To the right.
Moves to the right. Why?
Because it wants to reach equilibrium.
True.
It's a low concentration.
And it's out of balance. It has a concentration gradient. That's all the chemical force is. The chemical force is the concentration gradient, where things want to move from a high concentration to a low concentration to reach equilibrium. All right. And as we didn't mention, but you should easily be able to tell me, what would equilibrium be at this point for this beaker?
Um,
37.
There you go. Liz has got it.
Oh,
45% on each side, right? Add them together. That's 90. Split them in half. 45% on this side, 45% glucose on this side.
okay.
And remember, at this point, is there no more movement of glucose?
There is.
Yeah, glucose still moves, but remember, at this point, there is no net change. So that means for every one that goes left, another one goes right. All right, we comfortable with that idea?
yes.
All right, let's take it the next step. The next step's where it gets a teeny bit trickier. Instead of 75% glucose on this side, I'm going to put 75% sodium chloride. On this side, I'm going to put 15% sodium chloride. And instead of just using a screwdriver, if I took a screwdriver and poked holes, then yeah, it would just simply go, we'd have 45% sodium chloride on one side, 45% sodium chloride on the other. But again, our cells don't have holes in them, they have channels. So let's say here, I had channels that were specifically sodium channels. Now, is there a chemical force on sodium?
Yes.
Yes. So which way are they going to move through this channel?
Gonna want to move to the right.
Yeah, and they're going to move to the right. Absolutely. That is their chemical force, right? Moving down their concentration gradient. Excellent. However, unlike glucose, sodium and chloride are charged particles. So instead of 75%, let's say there are actually 75 sodium on this side, and there are 75 chlorides on this side. And on this side, there are 15 sodiums and 15 chlorides. When one of these moves from the left to the right, we now have 74 sodiums on this side and 16 on this side. Notice we no longer have an equal charge on both sides. Notice this side has become slightly more positive. And this side becomes slightly more negative. Now, as we've mentioned, charged particles like magnets are attracted to opposites, are repulsed from positive, from the sames, right? That's our electrical force. The electrical force is the attraction to the opposite charge and the repulsion from the same charge. So if I had a beaker that had sodium in it, and one side of it was positive, and the other side of it was negative, which way would sodium want to move? Would it want to move to the left or would it want to move to the right?
The left.
Yeah, it's going to want to move to the left. It's going to want to go to where it's more negative. And that is its electrical charge. So as we know, the inside of the cell is negative. What was the membrane potential on the inside of the cell again?
Negative 70.
-70 millivolts.
Negative 70 millivolts. So notice what's going to happen here. Sodium may want to go down its chemical force to go into the cell. This is the inside of the cell. And since the inside of the cell is negative, sodium wants to go inside as well. So notice, being a positively charged ion, it wants to go inside. And so that's its electrical force. So notice for sodium, both its chemical force and its electrical force are driving it inside the cell.
So if this is different, this is when we would use the ATP to go against their gradient.
Well, the take-home message for this is sodium really. So if we go back to the idea of a cell, we make the cell. We know that the cell is negative on the inside. We know that there is a small amount of sodium on the inside. Outside the cell, there is lots of sodium. Outside the cell, it is positive. So the charge is the electrical force. And in this case, sodium wants to go in. The concentration gradient is the chemical force. And so it really wants to go in. So the point I was trying to make at that time is there are two factors that can influence an ion, its charge and the concentration gradient. And really the take-home message right now is that sodium really wants into the cell. It really, really wants into the cell. And as we talked about, we can use that to do work with our co-transporters. So that was really why we brought it up now when we did, is just to show you how much sodium really, really, really wants to get into the cell. And that's why we're able to use it as those co-transporters to bring stuff in. But remember, it only wants to come in because the cell is negative, because there's a small amount. So if more and more sodium comes in, the cell gets more positive, the concentration goes down, and it doesn't want to come in anymore. So that's why as soon as that sodium comes in, we need to pump it back out. So that was the point of the whole chemical and electrical at this point. Like I said, we'll use it much more when we get to the nervous system and the muscular system. But right now we were just using it to emphasize how badly sodium wants to get into the cell. and how we can use it to do work. All right, does that make sense?
Yes.
Okay, excellent. All righty. Next question.
Oh, I guess Mitch has the question. Never mind. I'll go after her.
I'm sorry?
Mitch has a question, I think, so I'll go after her.
Oh, okay. Here we go. Sorry, I didn't. Thank you. I didn't see that in the discussion. Okay, great question. So both lysosomes and peroxisomes are vesicles. So they're organelles, membrane-bound organelles. Oops. But they're very simple ones. They're basically a circular plasma membrane, and inside they contain proteins. So they basically look identical to each other. Lysosomes primarily contain hydrolytic enzymes. As we emphasized today, their job is to break bonds. And in particular, they play an important role in helping to break down unwanted or unused materials from inside or outside the cells. So, as we just finished talking about with mitosis, mitosis involves a tremendous number of special proteins that are used just in mitosis. So if you're a liver cell and you're not going to undergo mitosis again for a whole year, do you want those extra proteins sitting around inside your cytoplasm, taking up space, using up resources for something you're never going to use again for a very long time? No. So those proteins can be broken back down into amino acids to be used to make other things that that liver cell needs. Or as organelles get damaged or die, they can be broken down so those materials can be recycled and we can make more. Or in some cells, remember those cells we call phagocytes. Phagocytes are one of our types of white blood cells. And their job is to find some harmful virus or bacteria in your body. They engulf it and bring it inside, and then they use their digestive enzymes to break them up, destroying that virus so it doesn't hurt you. So basically, lysosomes, their jobs are they're the big recyclers. All right. Helping us to break down material from either inside the cell or outside the cell that we're no longer using, or is no longer functional, or is potentially harmful, and we want to destroy it. Now, the lysosomes are similar to that last part. Peroxisomes. Peroxisomes play an important role in helping to, let's say it this way. Helping to maintain the health of the cell. Some of this is basic stuff. They're going to be used to break down lipids, whereas our lysosomes are more involved in breaking down proteins. And things like that. But the other major role of our peroxisomes is to play an important role into detoxifying excuse me, the cell. This especially is true of things like peroxides. Peroxides are something that is very dangerous to the cell. Anybody know what peroxide is? Chemical composition of peroxide. Again, it always warms the cockle of my hearts when you guys don't know chemistry stuff, because chemistry is stupid. But two chemists walk into a bar. The first one says, "I'd like some H2O." Second one says, I'll have some H2O2. And the second one dies because H2O2 is peroxide. And it is toxic to our cells. So one of the things that our lysosome is able to do is to break it down into water, which is not toxic, and oxygen. It also plays a role in helping us to get rid of free radicals. Free radicals are things that cause peroxides to be formed and things along those lines. So our peroxisomes play an important role in helping to maintain the overall health by helping to detoxify, breaking down toxins, breaking down lipids, breaking down peroxides and things along those lines. All right. Now, the last thing I would say is, remember when we were talking about these on our models or on our charts, our lysosomes and our peroxisomes, when we looked at the model, there wasn't really any distinct difference between them because they're just basically ball-shaped organelles. So we came up with a rule. And what was the rule that we came up with?
Peroxisome.
What color are our lysosomes going to be?
Purple.
Purple, excellent. And what color are our peroxisomes going to be?
Yellow.
There you go. So on the models, the yellow balls inside the cells are the peroxisomes, and the purple balls inside the cells are the lysosomes. So that's how you'll tell them apart on a model or a chart. And then now we know their functions as well. Did that answer your question, Mitch?
Yeah, thank you.
Excellent. All right, Ava, I think you had a question then?
Yeah, I have a few. I have a few small ones. So
Okay.
I know you mentioned something about the lab exam being curved. Are both of them curved or just one?
All exams are curved.
Okay.
So both lab
And then when we're let's say for an essay question, you ask us to describe the mitosis process. Would you expect us to include the S phase in the beginning, even though it's technically not considered part of it? Okay.
No, if S phase, you were correct. S phase is a part of interphase.
Right.
So you certainly don't have to describe it. I guess if you were describing a chromosome and you were mentioning how it was made up of two sister chromatids, it wouldn't be a bad thing to mention that copy was made during S phase. But no, I don't expect you to, if I ask specifically, say again, it's one of those great questions. If I'm asking specifically for mitosis, then I just want prophase, metaphase, anaphase, telophase.
Okay.
Right? If I'm asking for the mitotic phase or cellular division, then I want mitosis and cytokinesis. If I ask for interphase, then obviously I want G1, S, G2. And if I ask for the cell cycle, then I would want all of those things, right? So again, just make sure that you are specific to the question. If I just ask mitosis, there's no reason for you to talk about S phase.
Okay.
Now, again, you can always give me more information. If the more information is fine, as long as it's correct. Where you get in trouble is when you give me information I didn't ask for and that information is wrong. Then I have to start second guessing your answers. And like I said, I won't do that. So yes, there's nothing wrong with putting additional information. More detail is always better than less. Just make sure what you're writing is correct.
Okay.
All right. And you have another question?
Yeah, I was wondering if you can go over the levels of structural organizations and proteins. I'm having a hard time memorizing it. So I was wondering if you could just go over it again.
4 levels. Primary, secondary, tertiary, quaternary. All right. 4 levels to the structural organization of proteins. All right. Primary is just simply the sequence of amino acids. right? So amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid 5. It's just that, the sequence of amino acids. Our secondary is where our hydrogen bonds form between local groups of amino acids. So as these hydrogen bonds form between these local groups of amino acids, each different section starts to take on a little shape. So like we talked about, some of these may involve things like the alpha helices. Some of these may be beta sheets, things along those lines. But it's local changes and they're formed by hydrogen bonds. Now, as we talked about, if a sequence of amino acids has 3 parts to it, and one is an alpha helix, and one is a beta sheet, and then a second alpha helix, each of these individual parts would be a secondary structure, right? However, when you put all 3 of those structures together, you have the full 3-dimensional shape of this protein. And that's the tertiary structure. Tertiary is basically made from all the secondary structures put together. And basically it's the 3'. This is the 3-dimensional structure of the polypeptide. However, remember, most proteins are made of 2 or more subunits put together. So while this yellow polypeptide is 1 subunit, as we talked about before, we may need also a globular subunit. We may need some triangular subunits. And when we put those subunits together, we now have a fully functional protein. And that's the quaternary. Our quaternary is when we put 2 or more polypeptides together to make a fully functional protein. And now we can add one more thing to this. Where does this part occur? Making the sequences of amino acids. If only I had learned like an hour and a half ago where I put a sequence of amino acids together to make a polypeptide.
Ribosomes?
Excellent. This occurs in the ribosomes. Where does the cutting, the folding, and the putting the pieces together to make the fully functional protein, where does this occur?
Golgi complex.
Exactly. Oops. The Golgi apparatus. No, it wasn't it. Apparatus. There we go. And the Golgi complex of the Golgi apparatus. Absolutely. So there you go. We were able to add one more piece to the puzzle. Did that answer your question? Excellent.
Thank you.
Aubrey.
Hi. I had my notes didn't really do me justice for tonicity. Tonicity, is that just referring to like hyperbolic solutions and whatnot?
Well, careful. So tonicity, remember, is that concept that we were talking about in relation to the importance of osmosis. Right? Where basically, as we know, water goes to stuff, or like we also talked about, we could think of it the other way, water moves down its concentration grader. So the environment that a cell is in is important for its function. So as we talked about, if we put a cell in a beaker, and it was an isotonic solution, what does that mean?
Means like there's an equilibrium of stuff in water.
Exactly. Basically what that means is the fluid around the cell has the same percentage of water and stuff as the cell. And you are absolutely correct, and that is vitally important for the function. We are going to be at equilibrium, equal movement of water into and out of the cell. Which is absolutely positively a good thing. However, as we mentioned, when you get rushed to the hospital because you are dehydrated, they don't take an IV, and with that IV, they don't just pour deionized water into your body. What kind of solution would that be?
Hyper?
Hyper?
Nah. No, hypo.
Hypo.
Yeah.
Hypo, remember, hypo means below. Basically what this means is the fluid, let's use the same wording I used before, around the cell has more water and less stuff than the cell. So when we put that cell in a hypotonic solution, what ends up happening? Is there going to be equal movement of water into and out of the cell?
No.
Sarah's right. In extreme case, absolutely. You got the right idea. Absolutely. Water is going to move into the cell. So we don't have an equal movement anymore. Water enters into the cell. The cell definitely swells. And as you mentioned, if it gets too full, just like a water balloon, it can pop. Of course, pop is not fancy enough of a word.
Lice.
So they use the fancy word lice. At the same time, when you're dehydrated, they don't take you and fill you full of salt water either, right? Like ocean water. Why don't they fill you full of ocean water? What type of solution would that be?
Hypertonic solution.
And again, let's keep our wording similar. fluid around the cell has less water and more stuff than the cell. And so what happens when that cell is put into a hypertonic solution?
Shrinks.
Exactly. Water is drawn out of it. Water is pulled out of the cell. And as we talked about, they often do these experiments with red blood cells, and they have a fancy name of that shriveling up.
Crenation.
Exactly. Crenation. Excellent. So when we talked about tonicity, really we were talking about two things. We were talking about obviously equilibrium and the health of the cells and the effect of osmosis, the effect of the concentration of water and stuff that surrounds a cell on the function of that cell.
Thank you.
Yep. Excellent. All right. Oh, did you have another question, or do you raise close? There you go.
I did.
I did it at the same time you did. All right, excellent. Any more questions? Like I said, if you have them, I'll happily answer them.
I have
Yes.
one more.
Oh, yeah, I'll be first, and then Augustina will go to you next.
Okay. I also was curious what exactly membrane potential meant.
So membrane potential just refers to what we were talking about before. When we talk about a membrane potential, what we mean is that the same way that there's an unequal distribution of ions, more potassium inside, more sodium outside, membrane potential just means that there is an unequal distribution of charges inside and outside of the cell. As it turns out, there's more negative charge inside the cell and more positive charge outside the cell. And just like concentrations, there is an equilibrium point. Now, again, is that equilibrium point the point where no more charged particles are moving?
No.
No. This is the point where there's no net change. Plenty of charges still move. But there's no net change. And that point of no net change, that point of equilibrium, we call the resting membrane potential. Because if you leave a cell alone, let it just rest and not do any work, it will stay at that resting membrane potential forever. And as such, what did we say the resting membrane potential was?
Negative 70 millivolts.
Now, one last thing. Of course, does that mean that every single cell in your body, the resting membrane potential is precisely negative 70 degrees, negative 70 millivolts?
No. Is there a range?
Yes, but remember, in this class, the sky is blue. So in this class, the number we are going to always use for the resting membrane potential is negative 70 millivolts. Okay. Excellent. Questions on that? All right, Mitch, I love your question, but Augustina had one first. Let me answer Augustina's and then we'll go through yours.
This one's kind of just a quick one, but I remember like one of the lectures you were going over general versus specific like body cavities that would be on the lab, like the test portion of the lab. Can you go over like one that would most likely be on?
Well, let's say it this way. Let's not necessarily worry about which one would be more likely, because they're all equally likely. And here's the other problem you run into. Yes, if I was writing your exam, then there would certainly be some that would be more likely than others. But remember, because we're online and because everybody gets a different test, I put all the possible questions into a test bank and they're randomly selected. So let's just talk about probably the most, the example that has the most steps, right? So let's describe the location of the heart. So we're going to think of in terms of the cavity location for the heart. If we were to start with the most general cavity, what would that most general cavity when we were describing the location of the heart be?
Ventral.
Excellent. Just out of curiosity, is there anything else we could have called it besides the ventral body cavity? There we go. All right, let's try to be a little bit more specific now.
Thoracic.
There we go. Thoracic. More specific?
Parietal pericardium?
No, I like where you guys are going. You're thinking of pleura. Remember, the pleura referred to the lungs. So it wouldn't be a pleural cavity. Was there a cavity specifically associated with the heart?
Mediastinum.
Okay, excellent. So within the thoracic cavity, we had the mediastinum.
Is that a cavity?
Yep.
Okay.
But inside the mediastinum, there was, and this kind of leads into what Mitch was asking about, a specific cavity formed by a serous membrane. And so what would be the most, there we go, excellent. So pericardial would be the most specific. So notice from the broadest to the most specific, we were able to identify all of the names for the cavities where you would consider finding the heart.
I have a question.
Yes.
Would like the visceral pericardium be the most specific?
No. What you are describing are the serous membrane associated with it. What I'm
Okay.
talking about are cavities.
Okay.
Because
So-
remember, the heart, and here, give me a second. I will do my best job of drawing a heart as specifically as I can here. Excellent. There is the heart. As you can clearly see, it looks exactly like one. As you know, that heart has a serous membrane that is in direct contact with the surface. And how would we identify that specific serous membrane?
By the organ it surrounds.
Exactly. So this one, so how would we call this membrane that I've just drawn here in purple?
Visceral.
Excellent. Visceral what?
Visceral serous membrane.
True,
Pericardium.
although we can be more specific. There we go. Augustina's got it. Pericardium. This is the visceral pericardium. Now, remember, this visceral pericardium is also in contact, which I'm exaggerating here, with a layer that forms a big space around the heart. So how would we identify this serous membrane? There we go, Crystal,
Parietal.
got it.
Okay.
And as we were just talking about the cavity or space in between these two layers, as we just mentioned, is the pericardial cavity. So notice the heart is located in that space, the pericardial cavity, that is formed between the two layers of serous membrane. This is a great lead in to exactly what I think it was Mitch who asked the question. If it's not, I apologize to whoever asked it. The identifying the different serous membranes. Now, we already know one way we're going to tell them apart. One way we tell them apart is by their location. As we mentioned, if they're in direct contact with the organ, how do we refer to them?
Visceral.
Visceral. And second, by the organ they're associated with. Organ or organs they're associated with. Right? Oh, wait, hold on, forgot. So if they're direct contact, we're visceral. If they're forming or lining the cavity, how do we refer to them?
Parietal.
Excellent. The second way is by the organs associated with them. As we just learned, if you're associated with the heart, we call you a pericardium. Give me another one.
Lungs.
Okay, excellent. What do we call the ones associated with the lungs?
Plural.
Pleura. Or plural would be the adjective describing it. Plural would be the noun. I remember the brain has a different, it has the meninges that protect it. So the brain and spinal cord are not covered by serous membranes. The serous membranes, remember, are only found in the ventral body cavity. So we have the heart, we have the lungs, and then the third one is basically in any organ in the abdominal pelvic cavity. And how do we identify those? Good guess, but not for the abdominal pelvic cavity. We just have one general name. What was the general name for the one associated with the abdominal pelvic cavity? There we go. Peritoneum. Excellent. So I haven't actually counted, but let's figure it out. Let's mix and match. Take a location, take an organ, give me an example of one of our specific serous membranes. Give me one of them. Okay, I'll go first because visceral pericardium. Describe that serous membrane for me.
It lines the pericardium membrane.
So what does that mean? So
it's touching the heart.
Exactly. It's the serous membrane that is directly in contact with the heart. Give me another specific serous membrane.
Parietal pleura.
Excellent. How would you describe that one?
The outer side of the lungs, not touching the lungs.
Not touching lungs, lining the thoracic cavity around the lungs. skip one because what else could we throw in here?
Parietal pericardium.
Excellent. And again, they don't have to be in order. It just helps me to remember them. How would you describe the parietal pericardium?
Outer membrane of the heart.
Excellent. Forming the cavity around the heart. Excellent. What else?
Visceral pleura.
Excellent. How would you describe that?
Membrane in contact with the lungs. For lungs.
Excellent. What else?
Visceral peritoneum.
How would you describe that?
Membrane directly on the abdominal pelvic cavity.
Well, the organs. So that would be
Right.
like the stomach or the liver or the small intestine or something like that. And what else?
The parietal peritoneum.
And how would you describe that?
The outer membrane of a lung, or not a lung, of the stomach or liver.
Yeah, basically it lines the abdominal pelvic cavity, the outer wall of the abdominal pelvic cavity. Excellent. And there you go. So apparently we have 8 specific serous membranes. Questions on that?
Would there technically be 10 because of the 2 lungs, the cavities are separate?
Ah, you got me. There is indeed 8, but you're right. We did mix 2. What are the 2 we missed? You had it. Give it to me. What were you explaining? Why did you say there was going to be 10?
Because the right and left lung have separate cavities and separate serous membranes.
There you go. Remember, the lungs have their own individual shrink wraps. So we have to distinguish the left and the right for those. So there you go. Indeed, we do have 8 specific serous membranes that we identify based on their locations.
And didn't someone say 10? Isn't there?
Well, I think they said 10 because they were trying to add the 2, but I
Oh.
had cheated. I went straight from 4 to 7.
Okay.
So IUM. But remember, technically you would get a letter. So if you put EUM, you would still get full credit for that on the exam. All righty. There we go. Any more questions?
I have one.
Okay.
Could you possibly pull up like, I don't know, like a sagittal plane cut and like point out what exactly organs are inside of it?
Now you have to remember, any section that cuts the body into a left and a right piece is a sagittal section, right? So they aren't necessarily all going to show all of the same stuff. This is a horrible picture for that.
Okay, then maybe like frontal, frontal.
Well, no,
view,
I'm
would that
just,
be-
so here's where I was here. I was trying to bring up the picture where we actually did the planes of sections, but notice this picture is so small, it's really kind of hard to see what is in there for those kind of things. So what I would, like I said, it really depends on the plane of section. It's not something that there's just going to always be something that you're going to necessarily see in one of these planes of view, because there can be all of those different parasagittals. So if it was something more lateral, where over to the right side, for instance, you might not see the liver at all, although in most of them, the liver being the largest organ is most likely to be seen in the abdominal pelvic cavity. So I'm not sure that there's just necessarily one view you have to memorize for that. But again, we did talk about the organs and the organ systems, so it is a good idea to look at that illustration from your textbook that shows the organs in the organ system to have a general idea of where they're located, or like we talked about when we were talking about, yeah, here we go. So something like this, where it shows the general location of those organs, something like this could be something that could be useful as well. So clearly, if you were to take a sagittal section through here, or let's go parasagittal section through here, you'd see the liver, you'd see the stomach, you'd see the large intestine, you'd see the small intestine, you'd see the bladder, right? You'd see the heart. You might not see the lungs because we're not lateral enough, but obviously if we moved it more lateral, then you would see the lungs, but notice we wouldn't see the liver anymore. So I don't think there's necessarily just one sagittal section where you're guaranteed to see everything. Does that make sense? All right, any other questions?
I have one more. Sorry.
Yeah, sure. No, as I said, no need to apologize. Any information, like I said, the only bad question is the question not asked. And as I've said before, if you have questions, I'm sure other people have as well. And so since not everybody is necessarily as comfortable speaking up, I greatly appreciate those of you that are. So please go ahead.
So this isn't technically like on the material, but you said that we can do little keys on the side if we were to like abbreviate something. So we would just have to write the key once, and then we wouldn't have to keep like what do you call it? Like keep writing the whole word out.
So yes, that is true. But here's the way that I would recommend it. So first, you can't have one key for the whole exam. So for instance, if you're talking about DNA, then what you can do on the exam when you're writing out your essay question is the first time you write it out, you can write deoxyribonucleic acid. And then after that, abbreviated DNA every time after that. So you could definitely do that. Or either the very beginning of the question or the very end of the question, you could just have key, and then you could have all your abbreviations and have them all written out that way. So that would be acceptable. However, on the first essay question, you can't abbreviate DNA and have your key there and have it count for all of the essay questions. Now, is DNA likely to be a part of all of the essay questions? No. So in most cases, whatever you're abbreviating typically will just be for that one essay question and wouldn't be for any of the other ones. So it wouldn't make sense to have just some big monster key at the beginning for the whole test. Keep it specific to the questions. And then lastly, on the lab exam, since lab exam is all about abbreviations, there isn't likely an opportunity for you to worry about abbreviations there. So on the lab exam, it's usually best just to spell everything out. Okay.
And then the lab exams, like about how many questions are there?
This one I think has somewhere between 60 and 80. I haven't finished finalizing it yet, so my guess is it's somewhere within that range. And like I said before, be careful. Remember things like left and right. There is nothing I hate more than someone remembering that this is the antebrachial region of the body, and then still only getting partial credit because they forgot to put right. Don't get the hard part of the question correct and miss the easy part. So make sure you're careful on those things. All right. Mitch, go ahead.
Can you talk about what types of cytoskeleton are in like villi and cilia?
No, because I don't want you to worry about that. That was specific information we didn't talk about. Remember, we didn't talk about the motility of the cilia and how it works. You just need to know that microvilli are static, whereas the cilia are motile. Your book does an amazing job of talking about and describing the components of that. I'm not holding you responsible for that information. But if you're interested, that's a great place to go for it. Yes, Fatu.
Can we talk a little bit about the cell cycle, like the G1 and G2 phase a little bit?
Sure.
Yes.
So I absolutely, in interphase, there are indeed 3 stages, and they are G1, they are S, and they are G2. And I get it, it's a little bit confusing. S has a different name, definitely something different is going on there. And as we know, that is when we're getting the replication of the DNA. So that one's very simple and very straightforward.
And we're binding the 2 copies together, right?
Well, yes, when they're made, they're kept together. Yes. So when we
Okay.
make them, we don't just let them run random. We are going to have them. But again, remember at this point, it's still loose chromatin, right? We're not folding it up into chromosomes with sister chromatids until interphase. I mean, until the mitotic phase. G stands for growth. So we have growth 1 and growth 2. In growth 1, this is more about making the cell bigger. Because after all, one cell has to divide into 2. So we need more cytoplasm. We need to have enough endoplasmic reticulum. We need to have enough Golgi apparatus. We need to have enough mitochondria, so that when one cell physically divides into 2, both are going to have enough stuff to be functional. All right. Whereas G2, which is right before the mitotic phase, this is really about making all the special proteins that are needed. Right? Unlike my garage, which is half filled with Christmas decorations that get pulled out once a year, put up, and then put away for another year, a cell isn't going to house all the special proteins that it needs every time it divides. So when it comes time to dividing, there are a tremendous number of specialized proteins that are needed for this process. And so G2 is really about making those special proteins that are going to allow the cell to physically divide itself into 2, allow the cell to move the DNA around the cell so that it can become 2. So both are growth phases, both involve making stuff, but they're making slightly different stuff. And there's one more thing we have to keep in mind in this. Remember, in G1, we start making the second centrosome. And in G2 is when we finish making the second centrosome. And remember, at that point, when it is done, that is the trigger for the beginning of mitosis. Oops. All right. Was that on the Physio X or was that on the review?
It
I
was on the Physio X too, something like that. There was a question.
Okay, well, I remember the question on the review where basically it stopped the breakdown of the spindles.
Oh yeah,
fibers.
it was the review then.
Okay, so if you stop again, so if you stop the breakdown of the spindle fibers, remember the breakdown of the spindle fibers are how the two daughter chromosomes are pulled to the opposite side of the cells. Remember, they start as basically being held together by that centromere. And then when the centromere inactivates, you get those distinct V shapes as the microtubule spindles are being broken down and they're pulled to the opposite sides. So if you can't break down the spindles, you can't pull the chromosomes apart. And if you can't pull the chromosomes apart, are you going to be able to effectively divide this cell in half?
Nope.
No. Now, I
So
don't.
when that happens, oh, sorry.
I'm sorry. No, go ahead.
When that happens, does the cell just die after?
Basically, what would happen is.
That's what I guessed. I don't know.
Yeah,
I don't know what would happen to it.
So in reality, you've got the right idea. The cell would become non-functional. So at best, it's going to be able to sit there and do nothing. Now, if it's sitting there and doing nothing, part of our body defenses may come and find it and destroy it because it's going to recognize it as being different, abnormal, and we want to get rid of abnormal things. Or sometimes what happens when a cell gets abnormal like that, special chemicals are released that trigger it to break itself down and kill itself. There's a fancy name for that. Anybody know the name of that, where we get a cell to undergo a programmed cell death where it basically kills itself?
I think it starts with an A. It's like
apoptosis. There
yeah.
you go, apoptosis. So often it can trigger something like an apoptosis where it would be
but it would never be it would never be like recycled. It would just always.
Well, I mean, once it dies, those components are released into the interstitial, and so sure, it's sodium, it's phospholipids, all those things would be picked up by other cells, because when it's destroyed in the tissue, it's going to be absorbed by the stuff around it. But yeah, no, it won't be functional anymore. Fatu, did I answer your question?
Yes, you did. Thank you so much.
Excellent. Aubrey, did you have another one?
Yes. For peripheral membrane proteins, are they always attached or affiliated with the integral membrane proteins?
No, often, but they don't have to be, no.
Okay.
So you've got the right idea. They are not directly attached to the phospholipids. They're not embedded or one or the two layers, but obviously something has to keep them close. So in most instances, that thing that's going to keep them close is another protein that they'll be connected to, but they don't have to be. There are some that can be held in place by other components. So it's not a part of the definition, if that makes sense.
Okay. And I had a question about contralateral and ipsilateral.
Yes.
For that to be represented on like a diagram, would it just be like an arrow kind of showing like movement?
Not necessarily. So, all right, let's pretend that's anatomical position. If I were to point at the left foot and the right hand, the right foot and the right hand, and ask you to identify their relationship to each other, right? Describe their relationship to each other. Certainly you could say, one is superior to the other or something along those lines, but you would also say that they're ipsilateral as well. They're both on the same side. So yes, like we talked about, it is more commonly used when we are referring to things like nervous pathways where information is coming into and out of the spinal cord, or when information is going into the spinal cord and crossing to the other side. But we can also talk about them in relation to things. So your right hand and your right foot are ipsilateral, whereas your right hand and your left hand are contralateral.
Okay, thank you.
Yep. In fact, now that I think about it, I think that would be really the only way you could describe the relationship between your right hand and the left hand. Because they're not medial, they're not lateral, they're not proximal, they're not distal, they're not superior, they're not inferior. Really, the only way you could describe, oh, I like that as a lab question. The only way you could describe the relationship between the right hand and left hand is to say that they're contralateral, right? They're on opposite sides of the body. All right, we got time for at least one more if you guys have it, or are your brains full? Aubrey, yes.
I had a question about phosphate. So I know with ATP, there's 3 phosphate, and when you break one off, that's the energy that you use. You mentioned that is it progressively more, well, it is because there's more negative charges. It's more difficult to attach another phosphate. Then would it be like exponentially less energy that you would get from taking off the next phosphate?
Yes, absolutely. ATP and ADP are both considered high energy molecules. But you are absolutely correct. The energy in this first bond, or really, I guess, second bond, is much, much higher than the second bond. So you are correct in that. So yes, when you break an ATP into ADP, you release much more energy from breaking that bond than when you take an ADP and you break it and you split it into AMP. It does release energy, but not nearly as much. So both technically are considered high energy bonds, but this second one between the second and the third phosphates is a much, much higher energy bond. And so it releases much, much more energy, which is why ATP is what is primarily used for the energy. There are some reactions that do use ADP in the body. I don't think we're going to talk about any of them in this class, but if you take a molecular biology or maybe even in micro, you may talk about some instances, but because it is a such much lower energy bond, ATP is the primary energy that we're going to use and talk about. Yeah, I'm trying to think. I don't think there's any example in 430 or 431 where we will use ADP as our energy molecule. So it can be. There are some places it is, but nothing we're going to worry about.
Okay. And then one question about testing. Do you think that we would be able to like still test in the ARC library or anywhere on campus?
I don't believe the library is an option, but I believe my understanding is that, I don't know. My understanding is that the computers at the library don't have cameras, so you wouldn't be able to use Broctorio there. I believe some of the computer labs on campus may have cameras that would allow you to be able to do that, but you would need to check with the specific computer rooms to see if that's something that they're compatible with. I don't inherently have, there's nothing in the way that I've set it up where it would be a problem. You just have to make sure that they're capable of accommodating you. So you'd have to check on the computer lab. You would have to probably call or speak with some of the computer labs to see if that's something that's possible. Now, if you're a DSBS student, then yes, they have specific rooms with cameras where you would be able to do that and they would be able to proctor it. But But again, I think those are specifically for the DSPS students. But if you require that kind of accommodation, that is something they can do for you. Although I'll be honest, I'm not sure if they could have that set up with you for you, and by you, I don't mean you, I mean anybody, in 5 days. So, but that would be something you need to check with them on. So I think those are your options, but I'm pretty sure the library, A, is too loud, but also I don't think those computers have cameras. So I think that's a problem. You couldn't do Proctorio there.
Okay, if I brought my laptop, would I be able to do that, do you think? I mean, I'll contact them.
Yeah, I think you would need to check with them. I mean, I know there are study rooms, so maybe it'd be possible to check out a study room where you could be able to do something like that. But again, I think that would be something you would, because it'd be nice to be on campus and be able to use their interweb. But I don't have any place where I can accommodate a student that way. And so you would have to talk to either, like I said, one of the computer labs or the library or DSPS. Those would be the 3 people that you would contact to probably get that kind of accommodation. They might have something like that in place for exactly this type of reason. I'm not aware of it, I'm afraid.
Okay, thank you.
Yep. All right, last call. All right, I think our brains are then full. I will remind you, I have, oh, did you have one more?
I do actually.
Oh, yeah, no problem. Go
So
ahead.
both the back of your hand and the top of your foot is named the same thing, the dorsum.
Well, what are your toes and your fingers called?
Phalanges. Yeah, you're right.
So the bones actually are the phalanges, but the digits, they're referred to as digits. So yeah, there is some similarity in those. Absolutely. Yep. All righty. Excellent. I will remind you, I do have office hours right after this. So if you have more questions, please come to my office hours or email me. I will happily respond to that. I will be around this weekend to answer any questions. And then I do have class tomorrow, which means I have office hours from 11 to 12 tomorrow, or at least 11 to 11:45 too, because that's when I have to leave to go get that class set up and ready. So if you have questions, you can reach me there as well. Otherwise, study hard, do well, vote on Tuesday, and I will see you in class on Wednesday. Good luck. Do your best on the exams. Read the questions carefully. Hold on. Let me get on my soapbox one last time. Read the questions carefully. People often lose points, not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. All right, now I'll get off my soapbox and you can ask your question. What was your question?
I was just going to say a comment. I know there was a lot of people at the beginning of class saying that they wanted to do like a study group or whatever. And I was just going to say, if anybody wanted to reach out and do that, to just comment on your discussion page, the water cooler or whatever.
Yep, absolutely. It's a great place to asynchronously be able to coordinate with people to figure out a good time to meet. Again, you guys can use Zoom. You guys can meet in person, whatever it is that you guys need to do, or just like I said, use the discussion board to communicate asynchronously. But absolutely, as I've mentioned, 2 things. Your grade in this class doesn't affect anybody else's. So if 2 people work together, they both gain and benefit from that. So it's not a competition. You're encouraged to work together. And I often believe that one of the best ways to learn something is to teach it to somebody else. So when you're working together and communicating together and helping each other, as long as you're, not talking about what's happening on, the Kardashians or anything like that, as long as you're being productive with the time, then by all means, it absolutely is something that is great and beneficial. So I strongly, strongly encourage it. All right, guys, have a good day, and I will see you next week.
Thank you.
Thank you.
Bye.